All right, good morning and welcome to the 2021 FDA Science Forum. I'm Mike Kaczynski and I will be helping running both of our sessions today. We have session 5, Advancing Products Based on Novel Technologies, and session 6, Medical Countermeasures, Infectious Disease, and Pathogen Reduction Technologies. Both sessions are running on YouTube and they're also on our media site website. Please refer to the flyers and all of that for the schedule. Also, we do have a poster session running also simultaneously during breaks. So at this time, I'd like to introduce you to your today's hosts in each room. In room 5, Sharon Watson, and in room 6, Rakshara. Go ahead and take it away.
Thanks so much, Mike, and good morning, everyone. My name is Sharon Watson from the Office of Scientific Professional Development within the FDA Office of the Chief Scientist, and I'm pleased to welcome you back to day 2 of the 2021 FDA Science Forum. The theme for this year is Science as the Foundation for Protecting and Promoting Public Health. Members of cross-agency science forum working groups have volunteered countless hours over the past year to organize this forum. Thus, I'd like to take a moment to give special thanks to the Science Forum Planning Committee and various work groups, all of our presenters, the CDRH Studio, FDA's web team, our AV team, Catherine Ng of Orsi, and Mike Kaczynski, who you just heard from, and the Citizen Staff College for all of their assistance for this platform. Today, we'll have 4 concurrent sessions that present the amazing diversity of the scientific research at the FDA. Please refer to the Science Forum's website for the webcast link to your preferred sessions. The 2021 FDA Science Forum virtual poster sessions, as mentioned, are also exhibited on FDA Science Forum's website. The posters are available for download to all FDA Science Forum participants. In addition, the audience will have the opportunity to email their questions directly to the designated poster authors from May 26th through June 9th, so you'll have plenty of time to view those. As mentioned, this event will offer continuing education credit for viewing the various presentations, so please view the FDA Science Forum webpage regarding CE for your preferred sessions. Lastly, you will receive a survey to evaluate the FDA Science Forum in the next few days. Please take a few minutes to complete the survey as we value your feedback to improve FDA Science Forums. Again, thank you so much for joining us today. Without further ado, we will begin today's event in this room with concurrent session 5, Advancing Products Based on Novel Technologies, where Beverly Lynn will be our moderator for this session. Beverly, the floor is yours.
Good morning and welcome to section 5. In this section, we will discuss the potential use, the limitations, and challenges of new technologies such as microphysiological systems, the microbiome, and the combination of both in regulatory science and advancing product development. Today, we will discuss in a series of talks the possibility of integration of these novel alternative models into potential regulatory environments that can be used to address unmet public health concerns, better models of human adverse response, as well as identifying and evaluating more reliable biomarkers for monitoring and interaction with the microbiome. It is my pleasure to introduce our keynote speaker to kick off this section this morning, Dr. Linda Griffin, Professor of Biological and Mechanical Engineering, Massachusetts Institute of Technology. After Dr. Griffin, we will hear from scientists representing all the centers of the FDA. Dr. Griffin.
Thank you so much for having me here today. I was asked to share with you some of the things we've been doing to overcome challenges in culturing super strict anaerobes that grow in the colon together chronically for days at a time with a healthy human mucosal barrier. And when we approach this problem, I've worked in the field of microphysiological systems since before it even started in the 1990s. We built models of liver and we built some other models. But the gut was a particularly challenging one for a number of reasons. First, what do we model? And functions of the gut depend intimately on the integrity of the mucosal barrier and its interactions with the immune system. So there's crosstalk not just between the microbes and the epithelia, but also the immune system and even the systemic circulation. So we started out thinking about particularly disease modeling, but these issues also arise in how drugs interact with the mucosal barriers, because many, many facets of metabolism, transport, et cetera, are dependent on the proper functioning of the mucosal barrier. So we think a lot about first what to model. For example, in Crohn's disease, there's need to model the influence of genetics, the influence of particular immune alleles, and so on. So we would like systems that allow us to come in and parse the human condition in a controlled way. If we look further ahead, we're not there yet, there's certainly a need to bring in the neural system. The enteric nerves play a crucial role in functioning not just of the epithelium, but also the immune component. So these are the kinds of questions and challenges that framed our approach in thinking about this. And of course, there's a huge force because of the lack of fidelity of common animal models to reproduce the human condition, particularly when it comes to the gastrointestinal tract. We eat different things, we have different microbiomes, there's different kinds of immune defenses in the mucosal barrier, and there are many instances, and this is just one, of when something appeared to be very effective and exciting in a mouse model, and then taken into humans, it turns out not to work. So we're motivated by how can we start to build these systems in a way that companies and academic investigators can use them to look at focused questions of how the microbiome, mucosal barrier, particularly in the colon because it's the most challenging, how do they influence therapies? And so here's our wish list of what we want to build in a model. We want, and this is the wish list, we're not completely there yet. Elements of this are present in various labs throughout the world, but no one lab has combined every single. I think we've addressed the very, very crucial element of the co-culture of the microbiome with a version of a mucosal barrier, and we're heading toward this complete. So let me take you through. Of course, we need complete anaerobic conditions on the apical side, and anaerobic is sometimes a relative term. There are many microbes, even C. diff and others, B. theta, that are often classified as strict anaerobes, but in fact, they can grow when there's a teensy tiny bit of oxygen. Other microbes, as the one I'll talk about today, Faecalibacterium falcinetti, which we use as our test case, is completely unable to grow if there's even a tiny bit. So we use the most extreme to test our system. And we want to have continuous flow so that we can build up a homeostatic model. And so a primary cell culture, KCO2s, are certainly valuable in many situations, but using primary cells in this situation is highly desirable. We would ultimately like to have a crypt lumen structure, so the normal architecture of the gut is the stem cells reside here and cells move up and then are sloughed off into the colon lumen, and we would like to get that architecture because the stem cells are driving the differentiated cells, and then this whole process will continue over time, and this is maintained by gradients of growth factors and metabolites. Ultimately, we want communication with stromal cells, immune cells, a vasculature, and lymph drainage. So this is the wish list, and I think we can accomplish this within a couple of years, actually having models that incorporate all of these. However, we're not all the way there yet in putting all the elements together. So I want to share with you some of what we have been doing, or where we are. So two huge areas of technical challenge are first the platform to meet those requirements of the total anaerobic flow on the apical side and aerobic conditions to nourish the mucosal barrier on the basal side. That's actually really hard, especially if you want a continuous flow in both places, so you can replenish the nutrients and have the spent cells and tissue washed out. We also have a lot of other features, such as being biocompatible, being able to sample, etc. Then there's the tissue engineering side and biological function. How can we make a device that accommodates the 3D tissue, and how do we make the 3D tissue that incorporates all of those elements that we want? So these are the things I'll briefly discuss now. So when we started this project, there were a number, and since then, there were a number of gut-on-chip devices that had been published in the literature, and we can go down and look at them and compare with respect to the materials they're made of, for example. We want materials that are compatible with using lipophilic compounds such as steroid hormones, for example, and lipophilic drugs. So having materials that don't absorb those are important. How the cells are seeded and matured, how long you can do the co-culturing, whether or not there's flow, what kinds of cells are able to be maintained, the kinds of species, and whether or not there's direct contact between the bacteria and host, because there can be concerns about overgrowth of bacteria. So these are all the kinds of questions that we set out to try to combine in one device. So a major decision we made early on was to focus on using transwell culture. This is a very common mode of culture for barrier epithelium of all kinds, and especially for the gut. We leveraged the incredible advance that Hans Klevers brought to the field in making organoids, and then others in the field, who I mentioned some of them here, have done tremendous work to show how you can build monolayers of mucosal epithelia from the colon and other parts of the GI system on a transwell. So you can take organoids and dissociate them and seed them onto a transwell, form a monolayer as shown here. So you see the monolayer here, the basolateral surface. This is from a publication by Nick Zakos. And then in many instances, if you want, you can add immune cells to the basal side of the membrane, and they crosstalk with the epithelia. These kinds of transwell systems are extremely well characterized in many labs, and investigators like using them. And so building a device that lets investigators take a familiar culture format that they trust and put it into a device that now allows them to culture with strict anaerobes was the direction we decided to take, with feedback from the community of users. And so the features that we are the key features of the system, which was really built by two wonderful postdocs, Yuzhao Wang and Bob Zhang, is here. And so let me take you through it a little bit. So there's a transwell with a monolayer of the mucosal epithelium shown here, and completely anaerobic flow enters and flows across the top of that epithelial barrier. We can introduce microbes into this apical side after the epithelial barrier and flow have been established. We could also introduce pathogens at a later time if we like. And so one feature of this, this is actually not drawn entirely to scale, because this gap in our device is actually 3 millimeters. And 3 millimeters is gigantic compared to standard microfluidic devices, which typically would have a gap of around 0.1 to 0.3 millimeters. So it's about an order of magnitude larger than most microfluidic devices that have been used. And why did we do this? We did this because it allows us to keep that mucus layer that's on top of very, very intact, and allows microbes to settle in and populate the mucus layer while we flow fresh nutrients across the top. So we bring nutrients in constantly all the time, completely anaerobic, flow it across the top so the microbes have a continuous replenishment of their food supply, as well as we can then wash microbes out and build a homeostasis. At the same time, there is circulation on the basolateral compartment to provide oxygenation and fresh media to the mucosal epithelial cells. We can also, if we want, add immune cells on the basal side of this. So this is the first implementation, a standard monolayer, which does not last more than about 4 days due to differentiation processes that are natural. We're in the process of building a 3D culture, but a standard monolayer is very valuable for studies at the present time. So let me show you some published work on how well, first of all, let me just remind you that the approach that we took also leveraged micromachining technology that we had developed for a liver application and commercialized. That's actually a device that has been, is in-house at the FDA, and the FDA has published on this liver device. And an advantage of this approach is that these hard plastics don't absorb lipophilic compounds. So if you're trying to do pharmacokinetics and look at fate of the drugs, then you have an advantage in not having them absorb to your device. And PDMS allows them to absorb. Another feature of this device is that the pump system was adapted again from technology used for the liver chip. It's a tiny, tiny microfluidic pump that allows us to, I'm just checking to make sure my phone is still on. It's a tiny microfluidic pump that allows us to have tiny, tiny little dead volumes so that you can squish the pumps and contain everything on one device versus having a lot of pumps wandering around and taking up space in the incubator and absorbing compounds. So that's a key feature that allows us to combine an anaerobic flow with an aerobic flow. Now, so this is what the device looks like in practice. I've shown you the schematic here, and there are 6 transwells together in a device that you set up and put in the incubator. This is a photo of it. When you maintain a colon monolayer in the device, it looks very healthy. You can see stain here. These are just immunostains. MUC2, these are making mucus. So that it's a healthy colon monolayer. And the features of the monolayer are such that we find a little bit of faster differentiation and repression of proliferation when we put cells from static culture into the GUMI device. So there's an upregulation of differentiation markers and downregulation of proliferation markers. This may be due to the fact that we have flow and a greater availability of nutrients for the cells in the device. Now that we've established that we have a healthy colon monolayer, we want to establish that we can grow a super strict anaerobe continuously with this monolayer and have some kind of biological function that seems similar to that in vivo in humans. And so if we look across the types of microbes that are present in the microbiome in the colon in humans, several of the very strict anaerobes, very strict is F. prausnitzii, erectile, and B. theta is a little bit less strict, are very dominant in the human microbiome, as you can see here. So they're highly present, and many of these have been implicated in disease processes, particularly deficiencies of some of them have been implicated in inflammatory bowel diseases. So many labs have reported, for example, on F. prausnitzii being implicated in ulcerative colitis and in other types of inflammatory bowel diseases. So across many different labs, this one seemed to be a particular microbe that, and one of the reasons it's implicated is that it is one of the major producers of short-chain fatty acids, particularly butyrate. So it ferments substrates that are present in the colon and produces butyrate, which can then act not only on the gut epithelia, but be transported across the epithelial barrier and interact with the systemic circulation and organs throughout the body. So we took F. prausnitzii, and the first thing we wanted to show is that we could maintain a barrier function and even grow the bacteria and get them to be homeostatic over time. And so this is just showing the number of microbes over time, and this is F. prausnitzii. So within 24 hours, we have what looks like a homeostatic number, and this is comparing to the CFU in the colon. And so then establishing that the bacteria are there by measuring them, we also then did some imaging to see where the bacteria are relative to the colon mucosal barrier. And so this is just a dramatic image of the colon mucosal barrier. The bottom of this would be the transwell. So this is after 3 days of co-culture, and you can see a dark layer here, which is very tight. In the colon, you have very tightly cross-linked mucus right at the surface of the epithelium, and then further out, there's more loosely cross-linked mucus where the microbiome takes up residence. And so this cloud of DNA here, so we've stained blue for DNA, this cloud here represents the bacteria growing in this lightly cross-linked mucus layer. And again, we don't have a thin gap, we have a huge tall gap between the surface of the epithelium and the top of our device, so the fluid can flow through gently and allow the mucus to sit there and host the bacteria in it. Now, importantly, and this is a little bit busy slide, so the important thing here is really looking at we're comparing static culture, the device without any bacteria, the device with bacteria, and then the culture medium that's fed across the apical surface to the bacteria. And we can see here, so we're looking at 3 short-chain fatty acids that are commonly examined, acetate, propionate, and butyrate. And butyrate is typically the one implicated in immune functions and inflammation. And we can see that only in the case of the presence of the F. prausnitzii bacterium, which is known to ferment butyrate, that we produce copious amounts of butyrate on the apical side. But important on the apical side, sorry, I'm getting used to using this coordinate. So the butyrate is here, acetate, propionate, butyrate. But in the basolateral medium, so this is on the other side of the epithelium, away from the microbes, in fact, we see copious amounts of butyrate when we have the fecal bacteria, fecal prausnitzii, fecal bacterium prausnitzii present. So the butyrate is not only being produced by these bacteria that are alive, healthy, well, in their anaerobic microenvironment, they are fermenting their substrates and allowing the epithelial cells to transport them across the barrier, as they should, into the basolateral compartment. In humans, these butyrate would then act on immune cells. It would be taken up into the systemic into the portal circulation, go to the liver, and impact the function of the liver. So this is moving toward a natural physiological state for this monolayer. There's also impacts of the fecal bacterium prausnitzii on immune modulators such as TLR3 and 4 and others. And this is still an emerging picture, and keep in mind we don't have immune cells present in this system, but there's clearly regulation of these TLRs, toll-like receptors that interact with microbes in the presence of the FP. We can also now go in and establish, and this is a manuscript in preparation that I thought worth sharing, we can establish some elements of the immune system, and here we focus on elements of the innate immune system, monocyte-derived macrophages and dendritic cells, which can be attached to the basal side of the membrane so that they interact with the epithelia and can serve as sort of sentinels for things that the microbes may be doing to the epithelia. And so we can also include in the circulation CD4 positive T cells, for example, to look further at, for short periods of time, 2 to 3 days, to look further at interactions with the immune system. And so again, even in the presence of these immune cells, if we look, there's no difference between the presence and absence of immune cells on the accumulation of a homeostatic number of bacteria density. It still looks the same as it did when the immune cells were not present, so there's nothing appearing to affect the bacteria, and the integrity of the monolayer is still quite robust. However, what we find is that there's a dramatic change in the baseline cytokine, chemokine profiles produced, particularly, of course, in the basolateral media, which you would expect because that's where the immune cells are. So we see a huge mostly an increase in various immunomodulators. And then also, though, in the apical medium affecting the epithelial cells, there's an increase in things like IL-17 and IL-9 that we hadn't detected before in the absence of the immune cells. So I've covered the platform. Let me finish up by describing some of the challenges in tissue engineering and then how we're trying to pull tissue engineering and platform advances together for a next generation that would be valuable in drug development. So again, we've focused on transwells so far. We leverage very strongly this organoid technology developed by Hans Clevers. And what he recognized is that the stem cell niche in the intestine maintains the homeostasis of the tissue. The stem cells reside in the crypt and they divide and the cells move upward and go into the lumen. And if you're in the small intestine, you go to the villi, and if you're in the colon, you go to the lumen. And these are maintained by growth factors produced by stromal cells here in the bottom of the crypt, so lymphs and other things. And so there's an intimate crosstalk. In maintenance of the tissue, there's also butyrate suppresses proliferation. So butyrate is going to make these cells more differentiated, but it doesn't penetrate down here. So there's a complex web of signaling that maintains this. A key advance in the entire field was the description by Hans Clevers and his postdoc Sato of how to grow these epithelial organoids in culture by taking advantage of driving stem cell expansion, putting cells in matrigel, basement membrane-like matrix that's extracted from tumor tissues. And so now the approaches for doing this have been described for mouse and human and IPS. There's a whole cocktail of growth factors used to expand the organoids in a stem cell state, and then you change the growth factors to allow them to start to differentiate if you're just growing the epithelia. So you grow just the epithelia, making up for the stromal cell production of growth factors by simply adding the growth factors to the medium. So labs all over the world use these approaches to expand patient-derived biopsies into tissue banks and do experiments. So now if we go back to our model, the thing that we want is, can we move toward growing a 3D structure that mimics features of this, including co-culture with the stromal cells and perhaps immune cells and so on. So to do this, we have over the past decade or so leveraged a common approach in tissue engineering based on polyethylene glycol branched polymers, 8 arms, as shown here on the left, that can be modified with small peptides that interact with cells or that bind matrix, a peptide cross-linker. These two reagents can be, and they're completely defined, so there's nothing, we know everything. We made it all in the lab. You combine these with cells and do a pH change that's very gentle, and you can make a beautiful 3D hydrogel like you see here that encapsulates cells in a very healthy way. And so we did a lot of engineering of this system. The basic system is there, but how you combine these reagents together to make certain mechanical properties, certain permeability properties, which peptides do you use, which integrin receptors do you target. These are all things that take a lot of engineering design, and I'm showing you what we came up with. We have a, what I call a one-size-fits-all matrix where there are peptides attached to the polymer, and these peptides bind to integrins, but there are also peptides that bind to extracellular matrix like fibronectin or laminin or collagen 4 that are produced by cells, so that when the cell makes their own matrix, the synthetic gel now captures it and helps the cell assemble it so that it can remodel the environment so that it is the natural environment the cell would have in vivo. It is a very versatile system because it can keep both epithelial cells, which want laminin and collagen 4 in their environment, they produce it, and stromal cells, which have other types of collagens and fibronectin. It can keep both cell types happy and also immune cells as well. So a wonderful postdoc in the lab, Victor Hernandez-Gordilla, was able to do a semi-empirical screen of a parameter space of matrix properties, biophysical properties, biochemical properties, and he identified, and this is showing organoids from the intestine growing in a standard droplet format, most organoid cultures done in just like 3 microliter drops on the bottom of the dish. And these are organoids that are grown in a completely synthetic hydrogel. They were grown from individual cells to form these stem cell, these were grown under the so-called stem cell expansion conditions. And what's essential is this peptide, the Gophoger, which we know interacts at least with alpha-2 beta-1 integrin, but probably others. And this is an essential peptide interaction because if we mutate one of the amino acids that is believed to be involved in binding with the integrin to a different one, just that one mutation to abolish binding, we lose the activity of the gel. So this is some evidence that it's working that way. So when we set this up, we screened it with a huge number of different donors, including mouse. Mouse is very easy to grow compared to human. And again, did a semi-empirical screen to arrive at a formulation that supports the emergence of Panna cells. They're characterized by this lysozyme, and they support stem cells. So we haven't definitively tested for stem cells. Those tests are rather hard, but this green indicates this cell next to the Panna cell is proliferating. So there are many features of these that suggest that we do have the normal kind of organoids that you would get in Matrigel. We're right now, this is work not published but ongoing by a student in the lab, taking these into a morphogenesis situation so that you could, in a somewhat straightforward protocol, allow cells to undergo a transition, if they're at the surface of the gel, into a crypt lumen structure. And this is very, very early days, but we're leveraging principles of morphogenesis, and again, biomechanics play some role in this to try to get these structures. And it turns out there's very important features that the stromal cells can play in helping this morphogenesis process. And this is very much a work in progress, but we're moving quickly. We collaborate with David Bro, and Victor Hernandez continues to work with us on this. So this is, we're moving toward allowing us to generate on a transwell in situ a true 3D structure containing these other cell types in the 3D structure. Now I want to finish up by just mentioning that a transwell is useful for now, and we are commercializing this device. Since I made the slides even, the company that has produces the liver chip is now going to start producing this device so that others in the lab can use it. We're in the process also of building an industry consortium so people can start to do their own experiments and test how microbes may be interacting with drugs or the microbes may be influencing biology themselves. But where we're headed is to combine the mucosal barrier with wonderful technology from my colleague Roger Tam, who pioneered approaches to build microvascular networks in microfluidic devices. So what's shown in the movie is this is a microvascular bed built in a gel in between 2 microfluidic channels, and the green were immune cells that are moving through the channels. We have recently translated all of our microfluidic technologies into a single platform that we're commercializing for use in this application so that we could ultimately marry the GUMI device I showed you with simple devices on chips. So I'll end there and thank my wonderful, wonderful collaborators on this project, primarily Dave Trumper, who's in the mechanical engineering department, mechatronics, who led development of these, and Rebecca Carrier, a wonderful collaborator from Northeastern, whose lab focuses on nutrition and drug uptake in the gut. So I'll stop there.
Thank you, Dr. Griffin, for the exciting and inspiring presentation, not only addressing the challenges of these systems, but ending with the development of a platform. Our next guest will be Dr. Daniel Tedeschi from the Center for Veterinary Medicine. He will be further talking about advancing regulatory science through organs on a chip. Daniel.
Thanks, Beverly, for the introduction. Linda, that was a great talk. And for the next 10 minutes, I will give you a high-level overview of why we as a center are interested on the organ-on-a-chip technology and its potential to advance regulatory science in support of our center's mission. The effect of drug residues in animal-derived foods on the human intestinal microbiota is an important human food safety endpoint of concern that needs to be addressed during pre-approval evaluation of drug products intended for use in food-producing animals. This concern has been regularly addressed using in vitro and in vivo test systems. In the next couple of slides, I will discuss the limitation of existing test systems and what organ-on-a-chip potentially offer as an alternative to existing methods. Insight into the microbiome and its interaction with human hosts and their chemical environments can be obtained by using diverse non-human models. Non-human models can provide insight into the molecular pathways, physiological process, and microbial response to chemical stimuli. One example is animal models. Animal models are widely used to investigate the human microbiome for several reasons. It's much easier to manipulate animal models than human subjects experimentally. Animal models allow the careful control of experimental variables and reproducibility that's often important in human studies. There are, however, important caveats when we use animal models, including the salient difference in anatomy, physiology, diet, and microbiome difference between human and animal model. The other non-human model used to study microbiome-drug interaction is an in vitro system. The current in vitro system are either batch culture, with or without host interaction, or continuous culture without or with host interaction. The top two panels shows the most common system lacking living intestinal epithelium that have been used to study the gut microbiota. The bottom panels display the four current systems that have been developed to closely simulate human gut microbiota interaction. Recent advances in tissue engineering, microfabrication, and stem cell biology have enabled the development of sophisticated systems like organ-on-a-chip model that can recapitulate the key microenvironmental characteristics of the human or animal organs and mimic their primary function. I just want to give you a very brief definition of organ-on-a-chip. Organ-on-a-chip is a microfluidic cell culture system that recapitulates the in vivo tissue and organ level structure, function, physiology, and pathology in vitro. It could benefit humans and animal health, and it has a potential to lead to a paradigm shift in drug development, toxicological screening, personalized medicine, disease modeling. In short, it enables the creation of more in vivo-like in vitro models, and it has a potential to reduce and refine the use of animals in research. Microphysiological systems have been developed to model a broad spectrum of organs from heart, kidney, liver, to lung, gut, and many others to understand human organ development, disease pathology, and facilitate drug discovery. Broadly, we can divide this approach into two. The first being single organ platform, which integrates one specific type of tissue or organ and provide relevant microenvironmental context. For example, this figure shows intestinal microenvironment. It includes the 3D architecture of the intestine, the mechanical cues, the peristalsis movement and the flow stress, and the biochemical cues, the microbiome and the immune cells. Depending on the cues included, it can be a relatively simple cell or organoid-based intestine-on-a-chip model with intestinal cells cultured in a 2D monolayer, or it could be a more complex one with the 3D architecture of the intestinal cells co-cultured with multiple cell types, including the gut microbiome. The second approach, which is a more complex one, is a multi-organ on a chip system. This involves integrating multiple associated organ chips in a single platform, which allows the study of multiple organ function in a systematic approach. As I indicated at the beginning of my talk, FDA considers the effect of drug residues in animal-derived food on disruption of the bacterial colonization barrier and increase in resistance bacterial population in its pre-approval evaluation of drug products intended for use in food-producing animals. In light of this, we are evaluating emulate intestine on a chip system as an alternative in vitro model for co-culturing of human intestinal and microbial cells, allowing targeted perturbation experiment, specifically focusing two endpoints, which are disruption of colonization barrier and development of antimicrobial resistance. We are also evaluating the ease of use of the system, the variability, the reproducibility, and limitation. We are also looking at the duration. What I mean by duration is the number of days that the system maintains the cellular structure and function and mimics the human intestinal environment. We hope to also develop performance standards for validating the model. And finally, the validated model could definitely help pharmaceutical drug sponsors to provide data on the potential effects of animal antimicrobial drug residues on intestinal microbiota. In terms of our approach, we are collaborating with Emulate and Johns Hopkins, and we are coupling the power of microphysiological system and the next generation sequencing technologies. So we are using Emulate intestine on a chip system. The system has two channels separated by a porous membrane coated with extracellular matrix, with endothelial cells growing in the lower channel and epithelial or colonoid, in our case, growing in the upper channel in the presence of physiologically relevant luminal flow and peristalsis-like movement. In addition, we will be culturing complex human microbiota in direct contact with epithelial cells and create an oxygen gradient to mimic the intestinal microenvironment. As Linda indicated, this is going to be a very challenging task in terms of maintaining the oxygen gradient that's necessary for obligate anaerobes. However, Ingvar and his colleagues at the Wyss Institute at Harvard recently demonstrated anaerobic intestine on a chip model that can sustain over 200 unique operational taxonomic units from 11 different genera, including obligate anaerobic bacteria. They have shown that the chip stably maintained a microbial diversity like the richness observed in the human intestine. And we know that organoid culture have limitation in maintaining the cellular structure and function for extended time and sustaining the co-cultured microorganism for prolonged time. To make sure the system is performing as expected, we will be monitoring the morphology, differentiation status, and membrane integrity using microscopic evaluation, immunoassay, and biomarker analysis. We will be using metagenomics and metatranscriptomics approach to elucidate the microbiome host interaction, as well as the microbiome host drug interactions. In closing, we hope that our research efforts help validate intestine on a chip model as an alternative in vitro model to support evaluation of new animal drug products. It could inform and direct pre-approval steps within and beyond FDA. To acknowledge Kathleen Wish, who's my team member, and my collaborators, Sylvia Pinero and Jeff Gilbert from Office of New Animal Drug Evaluation, who have expertise in regulatory science, Jean and Stella, who played a key role on the study design, Odile from CEDAR, and I would like also to acknowledge my collaborator from Emulate, Jordan, Ben, and Graciela, who are experts on Emulate flagship organ chip model for a variety of differential applications. And finally, I would like also to acknowledge Mark Danwitz and Nicholas Zakos, leading experts on the use of human enteroids to understand intestinal physiology and pathophysiology for their advice and providing the colonnade for the project. Thank you.
Thank you, Daniel, for that exciting talk and discussing the importance of these system in advancing regulatory science. Our next speaker is Dr. Sangeeta Hare from NCTR. She'll be discussing the importance of the microbiome as an additional criteria for safety assessment.
Thank you, Dr. Beverly Lynn Cook. I would like to thank the organizer for providing me this opportunity to discuss my research at the FDA Science Forum. In next 10 minutes, I will be providing you the research outcome of my studies and would like to make you think about whether such approaches are useful for making the decision tree for GI safety assessment. So the goal of our study is to define the microbiome if it is adversely impacted by the xenobiotic exposure. And for this, we use the bottom-up approaches using the in vivo model. And second, we would like to develop the translational approaches using the top-down approaches using various in vitro systems and ex vivo systems. So the research tools that are used in my lab are to study the xenobiotic, microbiome, and host interaction. So for the xenobiotic microbe interaction, we start with simple experiments like MIC determination using the in vitro microbial culture, then move on to the batch culture, then ex vivo system, and the animal model. Similarly, we also use the xenobiotic host interaction using the in vitro culture system, either epithelial cells or monocytes, macrophages, and other cell types, 3D culture model, then human intestinal tissue explant, and animal models. So why an additional criteria for the safety assessment is needed? Because it is not sufficient to say that interaction of xenobiotic or drug may cause diarrhea. We need to know who is there and if they are metabolically active, and also what they do there. And for this, we use genomics, metagenomics, and functional genomics approaches using the holobiome assessment. So in our lab, we are using several xenobiotics and use this test system. So today, I will be mainly talking about the arsenic, how this can be used as a model xenobiotic to assess the xenobiotic microbe-host interaction. So arsenic is a ubiquitous xenobiotic, and the regulatory agency has defined the levels of this into the common food as well as into the dietary supplement and also in the infant rice cereal. The most interesting thing is that arsenic, when it goes into the gastrointestinal tract, it interacts with the gut microbiota and epithelial cells, and it can become more toxic by converting into the various toxic species. And this also causes concern for the gestational exposure and the intravenous exposure. So to study this toxicity, we have collaborated with Dr. Don Doshi at the Division of Biochemical Toxicology. And today I will be defining the outcome of the two experiments. First is the adult exposure, in which mice were exposed with 3 different concentrations of arsenic, and the neonatal exposure. And the samples were collected just after 24 hours or the 48 hours after the exposure. So here in the result, you can see that the control is defined at very right in each of the graph. And here is the aerobic culture, anaerobic culture, and specific bacterial culture. And you can see that within 24 and 48 hours, there is decrease into the live recovery of the live bacteria. Similarly, then we look for the 16S sequencing, and here are various groups with the low dose, medium dose, high dose, and you can see the discriminant analysis showed a separation for different treatment groups. Then we went ahead and did the sequencing, and results are shown in the heat map. At the very left is the control, and you can see that all different experimental group has higher abundance or the lower abundance of the some bacterial species that are shown by the arrow. Same thing, we looked also into the developmental stage, and what we see here at PND21, there is a distinct population during the exposure with the arsenic, and it is also very different from the adult control. So next, we wanted to see what is the immune status, whether it is affected, and here is the snapshot, and we see that the high level of pro-inflammatory and anti-inflammatory cytokine secretion was affected. And we look for the functional analysis for the microbiome. Here is the snapshot of those results in adults at the top and postnatal at the bottom. So the bacterial species, those are affected, are mainly the bacterial species, those are involved into the detoxification of the arsenic. And the lower abundance of the bacteria was those were involved into the butyrate production. And butyrate has a very major role into the gastrointestinal homeostasis. So thus, we wanted to see what could be the long-term effect of the arsenic exposure in relation to the GI toxicity. For this, we went ahead and did the in vitro culture using the intestinal epithelial cells. And here at different passages, we were looking for the expression of the genes. And here at the 14 passages, and these experiments are, by the way, still going on, the genes responsible for the progression towards the invasion and metastasis in gastric cancer were highly upregulated. So then we also wanted to see if the intravenous exposure is some different or the same way. So for this, mice were injected intravenously with the arsenic, and samples were collected 1, 4, 24, and 48 hours exposure. And the result here, when we compare the oral exposure to the intravenous exposure, were quite different. During the oral exposure, we found the bacterial species that were involved into the pathogenic bacterial invasion and were producing the immune responses, whereas when the intravenous exposure was done, there was disruption of the first protective layer and the epithelial layer disruption was seen. So next, we wanted to see if we can establish experimental model that can also be mimicked into the in vitro system. But before that, I would like to give you the results for how the oral exposure we see significant amount of signaling and inflammatory responses and no responses for the cell-cell junction, whereas IV exposure resulted in the downregulation of the cell-cell junction, whereas no major changes into the immune response related gene except after the 1 hour of incubation. And these were also correlated with the speciation of the arsenic that occurs to the oral gut microbiota. And we were able to see the several arsenic species, that is conversion of inorganic arsenic into the more toxic trivalent and tetravalent into the oral exposure, whereas in intravenous exposure, we were not able to see such species. So now we wanted to see if the in vitro system can be developed that can mimic the oral or intravenous exposure. So here the cells were cultured onto the transwell. Apical exposure mimicked the oral exposure and basal mimicked the intravenous exposure. And what we were able to see that there was indeed actually the changes into the TER when the exposure was done from the basal side. And same results were found for the expression of the genes that are involved into the cell-cell junctions. So next, our approach is to go for the translational research from the experience that we got from the lab animal model of how we can correlate it to the humans. So for this, we collaborated with collaborative human tissue network, and we obtained the biopsy punches or the tissue explant from them and culture it into 24-well transwell plate and do the exposure with the xenobiotics, and these experiments are ongoing. So with this, I would like to present you the decision tree for the evaluation. So here we can say that we start with the MIC determination, and then once if MIC is known, then it can further proceed for the animal study, and we can look for the abundance, we can look the metatranscriptomics, and we can do the metabolic profile. And if we find certain genes that are specific or bacteria specific, we can further investigate those. And similarly, we can do the host microbiome interaction also. So this contributes to the FDA strategic plan for advancing regulatory science. And we can say that this decision tree can form the microbial dysbiosis and intestinal toxicity be added as the endpoint for the hazard or preclinical safety assessment. And with this, I would like to thank my research team, and I would like to dedicate this presentation to Dr. Carl Seniglia, who passed away a month before. Thank you so much.
Thank you, Sagira. And because of time, we're going to press on to our next speaker, Dr. Zhuo Li from CEDAR, who will further discuss the microbiome. Dr. Li. You don't.
Sorry, can you hear me? Can you hear me now?
Now.
Oh, sorry. Yeah, I was on mute. Thanks, everyone. My name is Zhuo Li from the Division of Applied Regulatory Science, Office of Clinical Pharmacology, CEDAR, FDA. Today, I would like to talk about one project that we have been doing, studying the emergence of nosocomial opportunistic pathogens in the gut microbiome after antibiotic treatment, as reviewed by a mouse model metagenome analysis. First, some standard FDA disclaimer. So, what are nosocomial infections? Another name for it is healthcare-associated infections, or HAI. These are infections associated with stays in hospitals, rehab centers, and nursing homes, etc. And many of these infections are caused by opportunistic pathogens with different types of antibiotic resistance. Shown here are three famous or notorious nosocomial pathogens: MRSA, or methicillin-resistant S. aureus, A. baumannii, and C. difficile. I'll talk about them more in details later. Nosocomial infections have huge impact. If you are admitted to a hospital, you have a 5% chance of contracting an HAI. Every year, 1.7 million people got infected through HAI, and 98,000 people were killed by nosocomial pathogens. So, why this? When we study the emergence of these nosocomial infections, or where do these pathogens come around? There is a speculation that oral antibiotic therapies in healthcare settings are linked to a nosocomial infection. My apology, but there's a typo on the screen. It should be antibiotic therapies, not antibody therapies. This is because it's well known that antimicrobial agents can kill others but allow antibiotic-resistant bacteria to grow in the gut. So it sounds like a plausible mechanisms to induce the emergence of such pathogens. However, if you ask, do you have any evidence of emergence of these pathogens as a direct consequence of using specific antibiotics? As far as we know, there are some evidence for C. difficile directly induced by clindamycin. But that's it. For all other pathogens, the in vivo evidence to link them to specific antibiotics is lacking. This is because to prove this direct link, you need to exclude many other confounding factors like diet, baseline bacteria, other medications, etc. This is hard to do in a clinical setting. However, it is possible to control all these confounding factors in animal study, so we started using animal models. We first inoculated mice with uropathogenic E. coli to induce urinary tract infection or UTI. And then we treated the mice with 3 different common UTI antibiotics, ciprofloxacin, fosfomycin, or ampicillin. And then we sequenced all genetic materials in fecal samples from these mice through metagenomic shotgun sequencing. And then we mapped the reads to public databases of known sequences for taxonomy profiling, which means we want to know which bacterial species produce each sequencing read. So this sounds all straightforward, but when we started doing this, we were faced with a bioinformatics challenge. This is because many sequencing reads cannot be mapped to any known sequences in the database. This is due to multiple factors. The sequencing reads are short. On our platform, they are 150 base pairs in length. Also, there is incomplete representation in the database about known bacteria. Also, we are sequencing not from a single genome. We are sequencing from a mixture of genomes, maybe thousands of different bacterial species. Because of this, we found that especially for control samples, when the diversity is high, up to 90% of the reads cannot be mapped to any known sequences. So in a traditional study, you have to throw away all these reads, all this data. So we decided to try another way. We implemented a novel method to assemble short reads into longer genomic scaffolds. So instead of having 150 base pair short reads, now you can assemble them into longer genomic fragments, up to thousands of base pairs. Now you do mapping, and we found that the mappable reads increased from 10% before assembly to 80% after assembly for control samples, for example. That means now we only need to store away 20% of the data, not 90%. So what did we find from this new approach? Shown here are top 10 bacterial species identified through this assembly approach. Left column is control. Right column is ampicillin treated samples. As you can see, some bacterial species are marked in red. That's because these are known opportunistic pathogens linked to nosocomial infections. For control column, none of the top 10 species are linked to pathogens, but on the right, for ampicillin treated samples, most of the top 10 bacterial species are linked to nosocomial infections. I'm not going to go through every species, but I would like to point out several of them. For example, Acinetobacter baumannii, it emerged in military treatment facilities during the Iraq War, and hence it got a nickname Iraqibacter. Now becomes highly prevalent in civilian hospitals, and some people speculated that this is because of the transfer of patients from military to civilian hospitals. But anyway, now it's common. It can be found in every hospital. It's responsible for 19% of ventilator-associated pneumonia cases, so clearly a notorious nosocomial pathogen. So now let's take a look at another antibiotic, ciprofloxacin. Again, left column is control, top 10 species. Right column is top 10 species from ciprofloxacin treatment. As you can see, many of them are red too. For example, C. difficile, it may colonize human gut without symptoms. but it may expand after the use of antibiotics like clindamycin. And like I said, this is the only pathogen that has a direct link to a specific antibiotic before our study. Is responsible for 200K hospitalizations and 12K deaths in 2017. And another pathogen that's worth mentioning is chlamydia. It primarily causes eye, lung, or genital infections, but can potentially establish long-term infection in gut. And we found that after ciprofloxacin treatment, it's elevated. So the last antibiotic we tested is fosfomycin. Again, 2 out of top 10 pathogens. For example, bacteroides fragilis is commonly colonizing human gut without symptoms, but it is involved in 90% of anaerobic peritoneal infections. Also can cause bacteremia. It's increased after fosfomycin treatment, but none from control. If we put them together and if we do a statistical comparison, we can see that in this plot, the x-axis are different treatment groups, control or 3 antibiotics. The y-axis are number of reads normalized per million reads. If you look at panel A, that's about the pathogen Acinetobacter, calcoaceticus, and Bombardier complex. You can see after ampicillin treatment, there's a huge increase of the total number of reads. The reads are color-coded. The orange part are the reads associated with ARGs or antibiotic resistance genes. The blue part are reads not associated with ARGs, but we can focus on the total number of reads. The total length height of the bar is highly increased after ampicillin treatment. If we go to panel G, chlamydia trachomatis, after ciprofloxacin treatment, the total number of reads are increased a lot, and also some for ampicillin treatment. If you go to panel D, for bacteroides fragilis, after fosfomycin treatment, the total number of reads increased a lot too. So in a nutshell, we tested 3 antibiotics, and each one of them will cause a huge increase of pathogen reads. So in summary, we found that assembling sequencing reads into longer genomic scaffolds greatly increases the number of usable reads in the metagenome study. And we provided direct evidence for emergence of gut opportunistic pathogens associated with nosocomial infection after the use of all 3 antibiotics, ampicillin, ciprofloxacin, and fosfomycin. Remember, we only tested these 3. If we tested more, we may find more. Of course, whether or not this represents a general mechanism for the emergence of nosocomial infection is verification from clinical data, but as far as we know, we are the first one to report such direct evidence from in vivo studies for a number of antibiotics. That's all for my talk. I would like to thank the bioinformatics team and also the animal model team from my division, DARS, and also my division leadership, especially Deputy Director Ronnie Rouse, to give support to this project. And that's all. Thanks for your attention.
Thank you, Dr. Lee, for that great talk and discussing the importance of antibiotic treatment and the development of these pathogens. Our speaker will be Dr. Paul Carlson from CBER, who will talk about safety and effectiveness of fecal microbiota transplantation products. Paul.
Thank you. Thanks to the organizers for the invite and to everyone for watching these talks today. I'm just going to jump right in, as always, starting with the typical disclaimer. And then we'll get into the actual meat of the talk here. So a lot of my colleagues have talked about the excitement surrounding the microbiome and the potential for manipulating the microbiome already today. And you've heard multiple people mention C. diff. So these are just some older publications that I pulled for talks in the past. But C. diff has been mentioned, and here's one feces-filled pill will stop gut infection, specifically referring to using fecal transplants to treat C. diff. And it goes on, and I'll mention some other indications that people are interested in, but here's another one using potential for the gut microbiome to fight obesity. And then one of my favorites here, are you ready to swallow a pill full of poop? And it may be hard to see down at the bottom here, but at the bottom here, it talks about how fecal transplants may be a miracle cure for some of our nastiest illnesses. And as I'm going to go over in the next couple slides, this is the kind of thing that really concerns me in the context of fecal transplants and the microbiome excitement. This graph, I just pulled, I went to PubMed and I searched the term microbiome and then pulled the result numbers by year. And you can see that we have this explosion in microbiome-based studies just in the last 10 years, or 10 or 20 years, 10 to 15 maybe. And this really is leading to a situation where in many instances the hype is getting ahead of the science. Here are a few other websites I recently pulled off, and this demonstrates my concerns and the potential risks here. Do-it-yourself fecal transplant. Let's do this at home. Here's another DIY instructions. DIY instructions, at least this one mentions risks, not just rewards, it mentions both. And here's an article talking about how people are going to YouTube and Facebook for this information. And I just want to pull a couple, point to a couple quotes or a couple pieces of information from these. This one, if you needed a visual, this is going to be one of the best ones of the day for you. This is an MD talking in an interview saying if you've ever made a milkshake, you can do it. Really emphasizing to the general public how easy this is and not emphasizing the potential risks, which are potentially very significant. Here's another, this is from this article about, again, YouTube, Facebook, Reddit. This is where people are going to find their donors. This one was nice, but they had a bullet point here specifically saying do-it-yourself FMTs are not recommended by medical doctors. This is very true. There are potential severe risks of doing this yourself. I do want to point out this one comment at the top here. It says the procedure is only FDA approved in a medical setting to treat Clostridium difficile. That is not true. FMT is not FDA approved for any use or indication. I hear that a lot, and that's why I like to mention that when I give talks. FMT is allowed to be used under enforcement discretion for the treatment of C. difficile infections not responsive to standard of care. However, it is not, at this time, an FDA approved drug. It is still considered an experimental drug. All right. So just going to walk through the process really quick, and then I'll get into a little bit of data towards the end here of doing a fecal transplant for those of you who don't think about this every day like we do. We start out with a donor. The stool has to come from somewhere. That donor needs to be screened. So we need to know what to test these individuals for and how good our tests are for doing this screening. And what to test for is going to change over time, as I'm going to talk about in a minute. After we test the donor and that we confirm that the stool is okay, the stool needs to be processed. We have an arm of our research project looking at how that manufacturing process might alter the product, the viability of the organisms, and therefore the efficacy of the organisms. I won't have time to talk about that today, but we're really interested in understanding what happens when these things get exposed to oxygen or some of these other things that are commonly done in the manufacturing process. Once the stool is processed, you have a product. That product needs to be characterized. We need to have a potency assay. How do you measure the potency of this product? It can be complicated because these are complex and highly variable products. How do you measure the stability? How long can it be stored? And a big one there, how do you measure manufacturing consistency with such highly variable starting material? One of the questions that comes up frequently is, does route of administration matter? And I guess the answer is, in some instances, we know that it might, and in other instances, we know that it does not, but we don't really have the answer here. But there are two routes of getting into the gastrointestinal tract, and people are using fecal transplant products going in both ways. We have plenty of capsule-based products or products given by feeding tube, as well as colonoscopy and enema-based products. There's a lot of interest in using fecal transplant. There are a lot of clinical trials underway under IND with the FDA. These are some of the things that have been associated with a dysbiotic microbiome that people are interested in. And this is by no means an all-inclusive list. This is a short list. There are many, many other things that have been added or that could be added to this list. But if something has been associated with the microbiome, then people are interested in studying fecal transplants to treat that. All right, so I want to circle back now for the last half of the talk here to talk about our efforts in testing and in understanding both what we should test for and how good our pathogen tests are. I published this paper in Cell Host and Microbe just over a year ago now, February of 2020. It's nicely outdated already, and I'll tell you why that is. In part, you can probably guess based on what's happened since then. But this gives a good overview of the regulatory considerations for this product class, including a list of commonly performed donor screening tests that I won't have time to go through today. However, I'm going to go over a few safety alerts that have happened in the last few years, many in the last year, and these have been associated with changes in donor screening. So the first of these, we had two instances of transfer of ESBL producing E. coli strains from a donor to a recipient through fecal transplant. One of these two individuals actually died. This changed our donor screening requirements to now requiring not just one or the other, but both of these two things. So donors are now required to both screen for MDROs and to exclude individuals at high risk for colonization from their studies. More recently, we've had cases of enteropathogenic E. coli and Shiga producing E. coli transfer, and this has also changed our requirements. EPEC is now required. It was not before. And STEC has to be performed by PCR nucleic acid-based testing. It was missed by other methods. And finally, we come to the SARS-CoV-2 and the COVID-19 pandemic, and the fact that this particular virus has shown up in stool. And so we released a safety alert stating that stool donated after December 1st of 2019 should not be used in FMT products until appropriate screening plans can be put in place. I apologize for the noise. My neighbor's cutting his grass. All right, so very quickly in the last couple minutes, I'm going to go through what we have done in this process. We had a question: how can we minimize the risk of SARS-CoV-2 transmission in these products? We know the virus is in stool. The problem was there were no commercially available tests out there for detecting RNA in stool. So we sought to adapt or develop an assay to detect viral RNA in stool that could be used for this purpose. And we determined all of these things, which I'm going to go through briefly. All right, so very quickly, this is our assay. We started out with stool. We got stool that was donated prior to the pandemic from 2 different sources, 5 different donors. We got virus from a colleague at CBER, Tony Wang, inactivated virus that we could spike into the stool, and we did that. And then we went ahead, isolated the RNA, and performed the RT-PCR. We started out with the CDC EUA test because that was the first one that was available when we were working on these things initially in the pandemic. So here you can see our results. After much optimization and effort, we were able to develop a test that could detect virus down to 3,000 copies per gram in stool. This is comparable to what we see with other molecular tests for gastrointestinal pathogens, and it also came in at 600 copies per mil of the solution, the mixture that we made up. But I think the copies per gram value is the more accurate representation here. We did test storage conditions. I'll mention the freezing and I'll skip over some of the others, but we saw that if samples were frozen in various buffers, in PBS or Kerry Blair media, we did see a reduction in the signal. However, if they were frozen in star buffer or DNA RNA shield, we did not see any change in the stability of the RNA in those conditions. And we also went on to test this at 4 degrees and at room temperature, and you can see here some solutions are better than others depending on the conditions that you're using. Importantly, we were able to test this in patients. This was performed in pediatric patients because those are the ones that we could get enrolled at the time. And you can see right here that we were able to detect virus in stool of these individuals in about half of them, showing that this assay works for detection, but maybe not as a strong diagnostic test. And you can get more information on these studies in this paper that we published in Lancet Microbe recently. So in conclusion, we developed an assay that works fairly well. It has multiple potential uses. I've focused on FMT donor screening here today. We showed compatibility with storage. I didn't talk about the cross-lab functionality, but this was very important. We sent samples to another lab, and they were able to implement the entire assay on their own with just our samples. And we showed that this works in pediatric patients. And with that, I'll just acknowledge the people who did the work, in particular Mike Coriel, a postdoc in the lab, who handled the majority of the SARS-CoV-2 assay development. And with that, I will conclude. Thanks for your attention.
Thank you, Paul, for talking about the risk of these FMT products. Our next speaker will be from fifth name, Dr. Kristen Extra, who talk about microphysiological systems, regulatory research consideration, evaluation of a model system.
Thank you for the introduction. I'm very excited today to talk about our perspectives for regulatory research considerations with the use of microphysiological systems, using our evaluation of the 2-cell liver chip as an example. So microphysiological systems, or organ-on-a-chip platforms, are miniaturized physiological environments that are engineered to model organ-level responses. They do this with the flow of media over cells, stretch, and multiple cell types. And depending on these design parameters, chips can be used to study a wide variety of tissues, organs, and disease states. Additionally, connecting multiple organ chips together can be used to study multi-organ effects. So due to the growing use of microphysiological systems and a need for new alternative methods, the FDA Foods Program began a Creative with Emulate in 2017 to begin addressing questions for the fit for use of organ chips in a regulatory setting. Our laboratory was the first CISPAN laboratory to utilize the Emulate liver chips to answer these questions, and we received funding from DARPA to carry out this research. So we decided to begin with the liver chip because the liver is the primary location of drug metabolism. Drug-induced liver injury is a leading cause of acute liver failure, and liver toxicity is a leading cause of toxicity failures in the pharmaceutical industry. Additionally, the human liver chip has been shown to predict human toxicity that was not predicted by other animal models. So the liver consists of 4 main cell types, including the hepatocytes, which are the most abundant cell type in the liver, and they perform many of the metabolic functions of the liver. The liver sinusoidal endothelial cells, or LSECs, form the lining of blood vessels in the liver. The stellate cells are the fat-storing cells of the liver, and they're important in fibrosis. And finally, the Kupffer cells are the resident liver macrophages, or immune cells. And depending on how many of these cells are included in the chip, you can model the liver in many ways. So we decided to begin with the co-culture containing only the hepatocytes and LSECs, which will be discussed here. But we're also currently working on the more complex quad cell liver chip containing all 4 of those cell types. So here is Emulate's 2-cell liver chip. This PDMS chip consists of 2 channels that overlap, with the primary human hepatocytes seated into the top channel and the LSECs seated into the bottom channel in such a way that they're in close proximity to the hepatocytes. So the remaining components of the Emulate system are involved in the flow of media through the chip. The chips are they're connected to the pods, which contain reservoirs for both fresh and spent media for each of the 2 channels. Sorry, these are the pods. The pods are then placed into these blue bays and loaded into the Zoe, and the Zoe is what is responsible for controlling the rate of media flow through the chip. And then connected to the Zoe is the orb, which it supplies the power and CO2 necessary for the Zoe to regulate the flow rate. So in order to evaluate this system, we had 2 main objectives. The first objective was to evaluate the performance of the platform in a regulatory setting, and the second was to look at the quality of the data using compounds of known hepatotoxic potential. So looking at the performance of the system, we found that the Emulate system was easy to use, though there was a little bit of a training period involved. We saw that the critical components of the of the setup were consistently functional, such as flow rate and critical periods of experimental setup. And we were also able to develop standard operating procedures that were in line with regulatory accepted GLP animal studies, which includes documentation of all the critical processes and ensures data quality. In total, we developed about 40 individual SOPs with about 20 document recording forms. Of particular importance is our chemical evaluation, and this includes testing the solubility and stability of the compounds, confirmation of the dosing solutions, as well as a test material interaction analysis with the CHIP PDMS. To examine the quality of the data, we looked at its predictivity, sensitivity, and specificity, power, and variability using 8 compounds that were either known hepatotoxic compounds or non-hepatotoxic compounds at 4 concentrations based on their own hepatotoxic profile in order to elicit a wide range of hepatotoxic results. So in order to do this, we set up our experiments, which took about 2 weeks. During the first week, the chips that were set up, which includes activation of the chips, feeding of the cells, as well as preparation of the pods and connection of the chips and pods together. And then during the second week, the chips were exposed to the various test materials for 3 consecutive days. The test material exposure began on day 0, which is exactly 1 week after the hepatocytes were first seeded into the chips. We collected for several different types of analyses. We collected media from days 1 to 3 for a test material interaction with the chip material. Biochemical endpoints were analyzed from days 1 to 3, and the imaging analysis took place on the final day of experimentation. So going into our results, our test material interaction with the chip material showed that there was a wide range of interaction with the chip PDMS, from very high interaction with tamoxifen, and that 100% of the tamoxifen that we loaded into the chip was absorbed by the chip. Alternatively, we had very low interaction with the chip with PDMS, and that only 4% of this compound was absorbed by the chip. So with our biochemical and imaging endpoints, I'm not going to show any specific data because there's quite a bit of it, but instead talk about how useful these assays were for predicting toxicity, as well as what kind of considerations should be taken into account for their use. So albumin was our most predictive assay, and it was our best assay. Urea was a little bit less sensitive and less specific, and lactate dehydrogenase, or LDH, was predictive. However, due to the nature of this assay, consideration needs to be taken into account for the timing to make sure that it coincides with the lysis event. Similarly, with our imaging endpoints, we looked at nuc-flu, which marks nuclei, and this assay was a little bit less predictive in terms of toxicity, but it is a good indication of approximate cell number within the chip, which is a useful tool to have. TMRM, which marks mitochondrial membrane potential, was very predictive, quantifiable, and reliable. And caspase 3-7, which marks apoptosis, was similar to the LDH assay in that consideration of the timing needs to be taken into account to make sure that it coincides with when the apoptosis is happening. So I don't have a lot of time to go over the details of how we did our image acquisition and quantification, but we did recently publish a manuscript that goes into those details more thoroughly. But very briefly, we were able to capture images across the entire length of the chip, as well as through all the layers of the chip. So taking all of our results from the biochemical and imaging endpoints together, we determined that the liver chips accurately predicted toxicity for all of our compounds, except for tamoxifen, which again was shown to interact with the chip material. So after a collection of all of our data, we did a statistical analysis of the system, and here I'm going to compare this to our side-by-side 24-well plate studies, which had hepatocytes only in a dish, and this is the current gold standard for in vitro hepatotoxicity research. So here sensitivity and specificity are shown as area under curve values, and the values that are closer to 1 indicate higher sensitivity and specificity. So here, liver chips had high sensitivity and specificity, so it ranged based on compound and assay, and they generally performed similarly to wells. The notable exceptions are that the liver chips often had higher sensitivity and specificity in the, for usmic acid than the well did, whereas for tamoxifen, the well had higher sensitivity and specificity than the chip did. And all these highlighted portions are where one platform did it a little bit better than the other. So next we looked at variability both within components of an experiment, such as the Zoe and the Bay and between chips and wells, and between experiments, which is the experiment line. So here's sigma values that are less than 0.2 represent low variability, and as you can see, all of our values are less than 0.2, which indicates consistency of both within and between experiments. So finally, we did a power analysis to determine approximately how many cells or how many chips would be needed to see an effect of a specific size. And here a power of 0.8 or greater is considered acceptable. And for our imaging endpoints, we saw that an N of 3 was needed to see a large effect size, whereas for our imaging endpoints, I'm sorry if I said, I meant biochemical endpoints, an N of 3. And then for our imaging endpoints, an N of 4 was needed for a very large effect size. So to summarize, we saw that the liver chip system was easy to use, though it did require a bit of training, and we were able to develop many SOPs that were in line with GLP regulatory accepted animal studies. And when looking at the quality, we saw that the liver chip system was predictive, except in cases when the chemical interacted with the chip material. And in general, the liver chips had high sensitivity and specificity, high power, and low variability. And we recently published a manuscript that goes into this type of analysis in more detail with one of our compounds, diglycolic acid. And we're also currently working on a manuscript that is going to contain the results from the remaining 7 compounds that is going to look at that type of analysis as well. So to close up, I just wanted to talk about some of the things that we feel are important for consideration in the use of liver chips in regulatory settings. So first of all, we think that it's incredibly important to have good SOPs in place that clearly define all the aspects of the study. And I know I've mentioned this a lot, but most important is the chemical evaluation, especially the chip material interaction with the test materials, because this can have a direct impact on how much of the chemical the cells are seeing within the chip, as well as your interpretation of the results. Next thing to consider is which assays to use and when to use them. Not all of our assays were equally valuable in predicting toxicity, and incorrect timing can lead to missed results. Next, the cost is something to consider. Though liver chips may be a little bit less expensive than a full animal study, they're still quite expensive. The system, the chips, the cells, and all the assays needed to carry out the research all add up, and it's just definitely something to consider when planning these experiments. And finally, though we saw that the liver chips and wells tended to perform similarly on a statistical standpoint, we do think that with increased culture duration and addition of more cell types, more differences may become apparent. And that's exactly where we're moving forward with this. I already mentioned that we're going to be working on the quad cell liver chip, which has hepatocytes, L-sex, stellate cells, and Kupffer cells. And we'll also be looking at longer durations of exposure. So with that, I'd like to thank everyone on the liver chip team, especially Dr. Sprando, who is the PI of this project and planned it out from start to end. I'd also like to thank our FDA management and everyone at Emulate who has helped us out along the way. And that concludes my presentation. Thank you very much for your attention.
Thank you, Kristen, for the exciting talk and giving us the ins and outs of the use of the liver chip. Our next speaker will be from CBRH, Dr. Shelby Scoop. and talk about the evaluation of endothelial cells' responses to nanomaterials using a dynamic flow model. Kevin.
Thank you for the opportunity to present today. In this presentation, I will give an overview of nanotechnology and medical devices, as well as outline the challenges of biological evaluation of medical products containing nanomaterials and opportunities for the use of advanced in vitro culture models. I will then share data from one of our initial studies using these approaches on the evaluation of endothelial cell responses to nanomaterials using a dynamic flow model. Nanotechnology and medical devices includes a broad range of device applications as well as nanomaterial chemistries and morphologies. Medical products incorporating nanotechnology may include discrete nanomaterials such as metal and metal oxide nanoparticles and or immobilized nanostructures such as nanostructured titanium and electrospun nanofibers. These various types of nanomaterials are being investigated for medical applications such as diagnostics and therapeutics, including drug delivery and implants. Nanomaterials have been shown to influence a variety of biological responses. However, these responses are dependent on the cell and tissue type as well as the nanomaterial physical and chemical properties, such as size, morphology, and charge. Nanomaterial properties are also dependent on the physiological environment, including interactions with proteins and tissues in the dynamic system, making biological evaluation of nanomaterials challenging. Therefore, it is important to assess the nanomaterials in the context of their specific medical device applications. Due to the complex interactions between the nanomaterial physical-chemical properties in the biological system, evaluation of biological responses to medical devices containing nanomaterials is difficult, as traditional in vitro assays do not simulate the complex dynamic physiological environment. Additionally, evaluation of nanomaterials using traditional static 2D in vitro culture models can result in assay interferences or nanomaterial cell interactions that do not represent the clinical situation. Due to these challenges, safety assessment of medical products containing nanomaterials relies heavily on animal studies. However, there are ethical considerations, as well as these studies have high costs, are time-consuming, as well as having limitations such as limited disease models and potential lack of predictivity of human clinical responses. Based on these limitations and challenges, there are opportunities to utilize dynamic in vitro culture models to evaluate biological responses to nanomaterials in combination with animal studies for medical device development and safety evaluation. Microphysiological systems such as organ-on-chip technology has shown very promising potential for preliminary drug discovery and toxicity studies by providing more physiologically relevant environment, helping bridge the gaps between traditional in vitro cell studies and in vivo animal evaluation. Therefore, the use of dynamic in vitro culture models have the potential to simulate complex physiological environment and to provide more clinically relevant biological responses to nanomaterials. Our initial research in this area is focused on the use of an endothelium on a chip fluidic model to evaluate biological responses to nanomaterials under shear flow conditions that are similar to human vasculature, as nanotechnology is being increasingly applied for cardiovascular medical applications, and there is potential for nanomaterials in other tissues to enter the bloodstream. For these studies, we first conducted some preliminary studies to evaluate the impact of silver nanoparticles of varying size and concentrations using static in vitro testing to determine the size and concentration of nanomaterials, as well as the endothelial cell model for the development of our endothelium on a chip. We evaluated citrate-coated silver nanoparticles of 10, 20, and 100 nanometer diameter, as well as 4 human-derived endothelial cells, and evaluated cell viability and oxidative stress. These preliminary screening studies demonstrated that the endothelial cell responses were size and concentration dependent, with increased cytotoxicity and oxidative stress observed after exposure to the 10 nanometer silver nanoparticles. Furthermore, the study showed that the human cerebral microvascular endothelial cells, or HCMECs, were the most sensitive to the silver nanoparticles. Based on these preliminary studies, we decided to develop our endothelium on a chip using the HCMECs to evaluate the impact of exposure to 10 nanometer silver nanoparticles. For this study, we first characterized the silver nanoparticles using transmission electron microscopy to confirm the nanoparticle diameter of approximately 10 nanometers. Then, to assess the impact of the biological test system on the nanomaterial properties, we utilized dynamic light scattering to evaluate the hydrodynamic diameter and zeta potential of the in PBS and cell culture medium under static and dynamic flow conditions. These results showed increased nanoparticle size in cell culture medium due to interactions with proteins, as well as increased nanoparticle size under static conditions compared to dynamic flow conditions, potentially due to nanoparticle aggregation under these conditions. These changes to the nanoparticle properties are important to consider in the overall biological evaluation, as they can impact the cell interactions and resulting biological responses. To develop the endothelium on a chip model, we utilized a commercially available dynamic fluidic system to apply unidirectional flow with controlled physiologically relevant shear stress of 10 dyne per centimeter square. For our experimental design, first the human microvascular endothelial cells were seated in the flow chamber and incubated for 72 hours under static conditions to develop a confluent monolayer. They were then exposed to the silver nanoparticles at concentrations ranging from 0 to 40 micrograms per mil under the 10 dyne per centimeter square flow conditions or static control conditions for 24 hours. After the 24-hour silver nanoparticle exposure, endothelial cells and supernatant were collected for the biological assays. The endothelial cells exhibited significantly higher cytotoxicity under flow conditions compared to the static conditions. We also observed increased accumulation of the nanoparticles under the dynamic flow conditions, which was not observed in control studies evaluating silver nanoparticle interactions with the channels in the absence of the endothelial cells. Elevated lactate dehydrogenase or LDH levels were observed in endothelial cells under flow conditions after treatment with 20 micrograms per mil of silver nanoparticles. Similarly, nitric oxide levels were also substantially increased after silver nanoparticle treatment under flow conditions. Caspase-1, an inflammatory response initiator, and Endocan, an endothelial-specific inflammatory response marker, increased after treatment with 20 micrograms per mil silver nanoparticles, suggesting a possible inflammatory response-related toxicity. We further evaluated the endothelial expression of pro-inflammatory cytokines after exposure to silver nanoparticles under static and dynamic flow conditions using a multiplex assay. HCMECs exposed to silver nanoparticles under flow conditions exhibited increased pro-inflammatory cytokines compared to cells treated under static conditions, including IL-1 beta, IL-6, IL-8, and TNF-alpha. Our results show that the dynamic flow conditions affect the silver nanoparticle physical-chemical properties as well as the nanoparticle endothelial cell interactions. We observed differences in the nanoparticle aggregation between static and dynamic culture, as well as observed concentration and flow-dependent biological responses with increased cytotoxicity and increased inflammation compared to cells treated under static conditions. These studies have highlighted the impact of the dynamic physiological environment on nanomaterial physical and chemical properties, as well as the nanomaterial cell interactions and subsequent responses. However, further investigation is needed to determine the underlying mechanisms for these responses. To further develop the model and to expand our research into use of additional cells and organ models, as well as multi-organ models, including commercially available systems, as well as investigation of additional nanomaterials used in medical devices. Nanotechnology is expected to continue to impact the design, development, and manufacture of novel medical products. The performance and safety of medical devices that incorporate nanomaterials depends on the physical-chemical properties of the nanomaterials as well as the biological systems. Advanced in vitro alternatives have potential to better simulate the complex physiological environment compared to traditional in vitro culture models and to provide more clinically relevant biological responses to nanomaterials. However, currently there are significant limitations on the use of such in vitro models, including uncertainty on translation to in vivo responses. Significant research is needed to validate any advanced in vitro alternative models before broad acceptance in the scientific community and regulatory acceptance for their specific context of use. Although further investigation is needed, there may be advantages for the use of these advanced culture models for product development and biological evaluation of novel technologies used in medical devices such as nanotechnology. I want to thank the many collaborators on this project, particularly postdoctoral fellow Dr. Yijang Liu, who will be presenting a poster on this project during the FDA Science Forum. And additionally, we would like to thank our funding sources. Thank you.
Thank you. And our last speaker for this section is Dr. Kim Sung Sung from CBER. And he will be talking about microphysiological systems to assess the functional capacity of regenerative medicine cellular products.
All right, thank you so much for the introduction. So the regenerative medicine is the process of replacing or regenerating human cells, tissues, or organs to restore or establish their normal function. And the products are typically autologous or allogenic cells that are isolated from tissue biopsies. And it also includes tissue engineered products that are combined with biomaterials and scaffolds. And the field of cell therapy is really rapidly growing, as you can see from this chart. And the number of INDs with the cell therapy development program is rapidly increasing. And we also start receiving several license applications as well. And this is only with the cell therapy project. And our division receives a large number of gene therapy INDs and license applications as well. As you can expect, the regenerative medicine cellular products are very promising and have potential to meet unmet medical needs. But there are still several manufacturing and regulatory challenges that we may need to overcome. The cells are very heterogeneous, and there is patient-to-patient variabilities, and the source material has limited shelf life and limited sample volume. And there is also limited starting materials for any test method development. And more importantly, our sponsors use a wide range of manufacturing protocols when manufacturing these products. So, and cells are also exposed to different materials and environments during manufacturing. So it is highly possible that the manufactured cells are not identical to the cells freshly isolated from tissue biopsies. So there are critical needs to develop new methods and identify quality attributes that we could use to reliably predict the biological functions of manufactured products. And that is one of the objectives of my research program. And various bioengineering systems such as bioreactors, biomaterials, and microphysiological systems have potential to improve both manufacturing and characterization of cell therapy products as shown in this illustration. And bioreactors and biomaterials may provide better control over chemical and physical microenvironment to enhance the expansion of cell products and maintain their functional properties during manufacturing. And advanced in vitro systems like microphysiological systems can be useful to characterize cell products in a physiologically relevant condition and also identify therapeutically effective cell batches. So in my lab in CBER, we use various microphysiological systems such as organoids and microfluidic systems to develop and improve test methods for cell product characterization and also to identify product attributes that are predictive of safety and effectiveness. And I'm going to present two specific examples. So in this first example, we measured chondrogenic capacity of multipotent stroma cells using the cell aggregate shown in this top left corner. And we study a donor and passage dependency. So we tested 8 different cell lines derived from different donors. And then we used 2 passages that are passage 2 and 3 for early passage and passage 5 for late passage. So we generated this multipotent stromal cell MSC aggregate and cultured them for 21 days in chondrogenic condition, which was created by adding TGF-beta 3. And after the culture, we measured 14 shape and size features. And the first 4 images on the left are the representative images of aggregate of early passage MSCs at day 7 and day 21. And the bottom images are from late passage MSCs. And you can see that MSCs at different passages show the different growth pattern, where the cell aggregates from early passages showed some size increase compared to the late passage ones. And to look at the trend, we conducted principal component analysis and generated the score plot shown in the center. And we noticed that the factors associated with the principal component 1 were related to the size feature of the aggregate, and principal component 2 were related to the shape of the aggregate. So the 2 plots shown in the middle shows the principal component 1 value, which is basically a size feature of the aggregate. So you can see that all of the aggregates from the early and late passages showed initial size decrease from day 0 to day 4. And most of early passages MSCs, except the 2 cell lines shown in blue and green, showed size recovery after the first decrease. However, the late passage ones did not show any size recovery except the 1 cell line shown in yellow. And then we conducted histology to detect the deposition of cartilage-related extracellular matrix, such as sulfate GAG shown in blue and collagen shown in red. So as you can see that the cells that showed size recovery deposited higher amounts of extracellular matrix compared to the ones that did not show size recovery. We then looked at the correlation of size feature with the synthetic activity such as GAG and collagen production and chondrogen expression. And it was very interesting to see that the synthetic activity, but not chondrogen expression, correlated with the size features by day 7. So the take-home message from this study is that our high-throughput and non-destructive approach, which basically measures the size of the aggregate, could potentially serve as a tool to identify MSC lines that are more chondrogenic by day 7 instead of day 21. So therefore, this could really improve the manufacturing strategies for MSC-based cellular products for cartilage tissue repair. And in the first example I just presented, we looked at the direct differentiation of MSCs into cartilage-forming cells. But MSCs are also known to produce a lot of paracrine factors that could be beneficial for wound repair, immunomodulation, ischemic reperfusion. So in this second example, we study whether we can develop a new quantitative assay to measure the paracrine potential of MSCs to induce vasculogenesis. So in this second example, we use the compartmentalized co-culture platform to co-culture MSCs with endothelial cells to look at the MSC's vasculogenic potential. And like the chondrogenesis project, we look at both donor and passage dependency, as you can see from the configuration table. And we use lung fibroblasts as a positive control, since it is well known that the lung fibroblasts induce vasculogenesis. And the images shown in the bottom left are the representative images of endothelial cells co-cultured with different MSC lines and lung fibroblasts. And as you can see from the image, the extent of vasculogenesis was different between MSC lines. And since we are interested in developing quantitative assay, we are currently quantifying the extent of vasculogenesis using different algorithms. And as an example, on the right side, we have a bar graph showing average vessel length and the number of junctions. And then the first 2 bar graphs on the top show the donor dependency. So the first 4 graphs, blue, green, orange, red, are from different MSC lines, and we have lung fibroblasts in pink and endothelial cell monoculture in purple. And the bottom 2 shows the passage dependency, where blue is from passage 3 and green is from passage 5. So even though this is not a complete data set, these data suggest that vasculogenic potential of MSCs appear to be influenced by manufacturing parameters, such as the donor source and passage number. So we think that it is important to have a robust and reliable assay to understand the functional heterogeneity of the product and also the influence of the manufacturing parameters. So in my presentation, I showed 2 examples of how we utilize microphysiological systems to evaluate functional capacity of cellular products. And I think that the various microphysiological systems could be very useful in improving our understanding of cell-cell interactions, cell materials interactions, and also it may improve our understanding on the impact of the numerous manufacturing parameters. So with that, I'd like to acknowledge my lab members. These are wonderful, hardworking people. And also I'd like to acknowledge my collaborators and funding sources. Thank you very much.
First of all, I'd like to thank all of the speakers in this session 5 today, advancing products based on novel technology. I would like to say that we met our learning objectives. We discussed the potential utility and challenges of these new technologies. So in closing, I would like to just go over a few of the advantages of these systems. So in evaluating these in vitro microphysiological systems for organs on a chip, there are several advantages. These systems can represent major organ tissues in the human body, which can facilitate the assessment of biomarkers, bioavailability, efficacy, and toxicity of therapeutic agents or preclinical studies. Also, they can provide insight into normal human organ function and disease pathophysiology. They can bridge gaps between traditional in vitro cell studies and in vivo animal evaluations and further provide prediction of clinical responses for safety assessment. These molds employ the use of multiple cell types and flow media to mimic physiological stimuli. So advances in development of these molds using sophisticated integration of biology and engineering, as you saw in the presentation today, they have resulted in the development of molds for human micro interactions, as we examined in these presentations. We further discussed in this section the importance of the microbiome in relation to the microphysiological system. The microbiome is a key contributor in the metabolism of drugs, food additives, pesticides, herbicides, and other contaminants. There are knowledge gaps between the interaction of these systems with the host and the microbiome of these microphysiological systems. So therefore, increased understanding of the impact of antimicrobial drug residues on the human intestinal microbiome is needed, including the development of antimicrobial resistance, as discussed in some of our presentations today. These systems could improve the mimicking of human intestinal physiology and microbial complexity to address these gaps, as also shown in a number of the presentations today. However, there are challenges, as was discussed by a number of the scientists in their presentation today. One of the major challenges as we go forward with the system is the acceptance of these molds as new tools for use in regulatory settings. Furthermore, the validation and the qualification with these molds, will they produce results that are consistent? Also for the need for the development of standards for regulatory acceptance of these novel products, as we're seeing. So, the question that I will leave you with today, as we close out these sections, are these the tools for regulatory decisions in the future? Validation of these molds are currently being conducted not only as we discussed today, but throughout the country in a number of laboratories. Also in the various centers. We had representatives from each of the FDA centers discussing these molds technology today. therefore to better understand the usefulness of this technology. So, as I close out this section today, we did not have an opportunity for questions for our speakers, but I would like to further thank Dr. Linda Griffin, who kicked off this section for us as our keynote speaker from MIT. I think this was a very informative section, and therefore we will now end this section. But before we end this section, take our lunch break, we would like to encourage you. There are a number of posters in this section that will further discuss other models or other systems that was not discussed in our presentation. So please go and visit those posters section. Also, before, if you have any questions, please, you can contact me. I am Beverly Lynn Cook, who has served as your moderator for today. And my contact information is I am at the National Center for Toxicological Research. So as we continue to discuss and close this section today, I would like again to thank each of the speakers for their presentation. As I said again, the use of these models are great potential for regulatory science. At this time, I will like to further discuss the interaction of these models with the microbiome. And also, one of the presentations that was highlighted in our talk today, and it really focused on the fecal transplant and the use of these potential products without the approval of the FDA. And also, because of the COVID infection, I thought that presentation really brought home the use that these models should be evaluated very closely for use for potential diseases. They have a lot of potential use to be used. However, but consideration like the media, Facebook, and all of the information that sometimes is given to the public, it is important for individuals to really evaluate what is being shown in these media platforms. So again, I would like to say that advances in bioengineering and material science, as stated in our first talk by Dr. Griffin, is critical. And also it is critical for the development and the understanding of the various platforms that are being utilized in these microphysiological systems. So today we have discussed in the series of these talks the possibility of the integration of these novels, alternative models, into the potential regulatory environment that can be used to address a number of unmet public health concerns, develop models of human adverse responses, as well as identifying and evaluating more reliable biomarkers for monitoring toxicity, as well as the interaction with the microbiome. As I said before, there are a number of challenges of these new systems, as well as understanding them with advancing these product development. So, as I close this section out, again, I would like to thank all our speakers today that represented all of the centers. We were able to get this section and make sure that all of the scientists from all of the centers was represented to discuss these new technologies for advancing our products. Again, I would like to thank our speakers today. Also, it is important with our speakers for the responses for nanomaterials and use of these systems. We try to address every aspect that could be in addressing these new technologies. So I think we have, as I said before, met our objective for this section. We discussed in detail the importance of these systems as well as the challenges of these systems and the importance of integrating this knowledge into scientific communication with regulatory and research work. Again, as we close, the question I'll leave with you today is, are these tools for regulatory decisions in the future? Are these the tools? And therefore, as we continue to validate these modes, let's think about that, and also think about in the future, are these alternative models capable of replacing or assisting animal model studies for regulatory decisions as we journey through these sections. As we close, I would like to again encourage you, as we take this lunch break until 12 o'clock, there will be 2 concurrent sections in the afternoon. Please visit the poster exhibit. Again, thank you, and I hope you enjoyed section 5. No.
Welcome back, and welcome to the 2021 FDA Science Forum. As I said, welcome back from lunch. We now will be having our next two sessions, which will wrap up the FDA Science Forum, Session 7 and Session 8. Session 8 is Food and Cosmetic Safety, and Session 8 is Substance Use, Misuse, and Addiction. Just a reminder, you can check the agenda to find out which rooms you need to be. They're both being broadcast on YouTube and MediaSite. And with that, I'd like to introduce today's moderators. For Room 7, we will have Chad Nelson, and Room 8, we will have Marta Skowalski. And with that, take it away, both of you.
Thank you, Mike. Hello and welcome to the FDA Science Forum session on food and cosmetic safety, the role of innovation and technology. My name is Chad Nelson, and I am with FDA Center for Food Safety and Applied Nutrition. I have the distinct honor and privilege of introducing the distinguished speakers for today's program. First, we will hear from Dr. Callie Neal, Professor of Animal and Food Sciences at the University of Delaware, on health as a collaborative response to food safety risks. Our second speaker will be Dr. Susan Main, the Director of FDA's Center for Food Safety and Applied Nutrition, who will present on CFSAN's use of innovative science to address current and emerging public health priorities. Next up, we will have Dr. Dan Rice, the Associate Director of the Office of Food and Feed Laboratory Operations in FDA's Office of Regulatory Affairs, who will talk about FDA's support of recent foodborne illness outbreak investigations. Our fourth speaker will be Dr. David Edwards, Director of the Division of Animal Feeds in FDA's Center for Veterinary Medicine, who will present on what won't an animal eat, innovation in animal diets. And last but not least will be Dr. Luisa Camacho, a research biologist in the Division of Biochemical Toxicology at FDA's National Center for Toxicological Research, whose presentation is entitled Mind the Data Gap, Contributions of FDA's NCTR to Evaluate Cosmetic Safety. Following the presentations, Dr. Jeff Ward, Senior Science Advisor in FDA's Center for Veterinary Medicine, will moderate our panel discussion. I encourage you to visit FDA Science Forum's website and read the full bios of the speakers in the forum brochure. Now I'd like to invite Dr. Neal to turn her camera on and begin her presentation. Thank you, and I hope you enjoy our session. Dr. Neal?
Good afternoon, Chad. Good afternoon, everyone. Thank you very much for the opportunity to be here with you today. It's really quite an honor and a privilege. And I'm very excited to tell you about One Health as a collaborative response to food safety risks. This is the University of Delaware campus for agriculture and natural resources, my college. And I wanted to show you this picture because it really does showcase One Health. We have a 300-acre laboratory right outside our doors. And we have greenhouses for the plant scientists as well as food scientists. We have dairy and beef and sheep, and we have students everywhere, right? So this is really a wonderful showcase for how we think about collaborations within a One Health context. And many of you are probably familiar with One Health, and I'm going to tell you a little bit about how I see One Health in terms of making connections. We really are looking for the optimum health of humans, animals, and the environment. But I want us to think more about how we get to that optimum health, the connections that we impact in doing that. I think that's what I really want to tease out over the next few minutes. And a little bit of background, One Health is a historical concept. It's not brand new, but many people are using it. It did start in 400 BC with Hippocrates on airs, waters, and places, and maybe some of you are familiar with that. It has been used in referring to zoonosis, and those linkages were described by Rudolf Virchow, who was a prominent German physician in the 19th century. In addition to his medical career, he really advocated for the importance of improved veterinary education. And that's really where One Health has been for a lot of the most recent years in particular with veterinary schools. And the UC Davis Veterinary School coined the term One Health, using it as a way to think about collaborations to effectively cure, prevent, and control illnesses that affect humans and animals. And then you can see we've even broadened that scope a little bit. The CDC established an Office of One Health in 2009 and the USDA in 2012. Of course, Many of you are familiar with this growth in population and our need to find a way to better advance food security and food safety. One Health provides a way to be able to ensure the well-being of people, animals, and the environment. As we think about creative and collaborative problem solving, we really need to think outside of the box because we have limited resources, and so we need to work together. We cannot continue to feed people or animals in the way we are if we do not think about the impact on the environment. And as we are thinking about this connection between animals and humans, I would be remiss if I did not mention, at least briefly, what has been impacting our world for quite some time now and feels like a long period of time, I think to many of us. The origin of SARS-CoV-2 virus is still something that is being considered by many in different ways. But what we know about coronaviruses is that they frequent various animal types and animal populations. There are multiple spillover events that are possible. There are new spillover events right now that are being discussed by scientists around the world. And it's important that we understand that these spillover events may happen. And we need to think about how we're interacting and how we're surveying the environment. Really, that's that collaboration between these. Unique aspects of One Health have occurred over the past 15 months or so. And this is showing a couple of those thoughts about One Health. I attended a meeting by landscape architects where they spoke about the embracing a One Health concept for how we are designing new outdoor spaces, because these have become even more important to us over the past year. Here's an image up top from New York, and you can see the creative use of the outdoor space. And the slide image down there is one that I took a screenshot of during this meeting, where landscape architects were talking about One Health concepts in jungle environments, where some of these coronavirus spillover events or other viruses may be surviving and intermingling with multiple species of animals as well as humans. And so we can really broaden One Health to not just the veterinary schools anymore or the medical schools, but really to so many different contexts. And that's where I hope we continue to have this collaborative thought, right? And I love this infographic from the CDC. If I skip down here, to achieve the best health outcomes for people, animals, plants, and our environment, how do we get there? We have to have coordinating communication and collaboration. And without that, we are never going to achieve that. So we tend to stick in our silos. We tend to want to really address detailed questions of science, but we must approach these with a multidisciplinary and transdisciplinary approach in order to be able to achieve this optimum health outcome for all of us. And this is not just an issue in the United States. There are international collaborations on One Health. Here's some information from the OIE. And you can see here they are speaking directly to some of the zoonotic diseases of importance in both animals and humans. And many of the existing human infectious diseases are zoonotic, about 60%. Emerging infectious diseases are very high to be zoonotic disease, as we've seen with coronavirus. And so again, we have to think about how those interactions are happening and how we think about those collaborations to make health optimum. As a food scientist and food safety specialist, this is an image that I use in some of my classes, and you have perhaps seen this as well. And if you may be able to use the QR code there, otherwise you'll have access to this video. But the well-traveled salad is a wonderful representation of how we are One Health collaborative community when it comes to food production. We need to think about how we are producing food, transporting food, distributing food when we think about food production and food security, and making sure that we all have access to those delicious food products such as those shown here. And I know it's right after lunch or right at lunch, and so maybe some of you are enjoying this type of a global food product. So no matter what your role in science is, I believe that most of us, if not all, can have a role in terms of a One Health context. When I think about food safety, it really embraces One Health, and in fact, it may actually be the epitome of One Health. We must have adequate food and water supply to prevent disease. We must have that to be able to produce feed and food for animals and humans. And as a reminder, in the United States, about 1 in 6 Americans get sick each year from something he or she ate. So there is an important takeaway from this, in that we want to think about how we are producing food and who is getting what food to make sure that we can really, so we can make sure that we are connecting this collaborative aspect between humans and the environment where it comes to growing our food. And in fact, humans are really important because we play many roles in this, not only as scientists, but also as consumers. And when we think about emerging infectious disease in the context of ecosystems, that's also an important role when it comes to what consumers are asking of how we produce our food. And I think that consumers are really driving some of the change in ecosystems when it comes to food production. For example, more and more consumers want to eat local, which is a wonderful opportunity. It's a wonderful choice that we have, and in fact, something that you see and we all see across our food production supply chain are more and more choices. But consumers are really advocating for these choices, and they are driving change in the food production systems are responding to what consumers want. Some of that is not necessarily embracing the One Health concept, right? When we think about how important the optimum health is, we moved to production of animals in terms of indoor facilities, and you see here with swine and poultry, we moved to these types of environments because we wanted to be able to maintain quality, safety, and health of the animal. When consumers would like animals to be in a happier environment, to be outside and have access to the outdoors, that is not necessarily better for the health of the animal or the health of the human who will be eating that animal. In fact, we see an increase in chance of protozoan pathogens and various parasites, helminth parasites as well, and sometimes there's increased mortality. And we're also removing antibiotics from feed, which is not a bad thing, but we really need to think about what challenges come after we remove antibiotics from feed. And not just feed that was used here in poultry for growth promotants, because we've removed that for quite some time, but when we think about the health of the animals, how do we respond to that, and what happens to the environment? And so there's a lot more at play here than just making consumers happy and make them feel good about the food that they're getting. Of course, that's important as well. The intersection of One Health and food safety can include multiple issues, and some may be more familiar than others, and this can actually be quite complex interactions. If we look at these 3 different outbreaks that are all associated with Shiga-toxigenic E. coli, you can see here that we have a bison, ground bison, we have a plant product, and then we have an unknown source, right? And so they're all caused by the same organism, but very complicated issues that are very different surrounding each one. And certainly our pets are important animals to think about when it comes to One Health as well. And this is a showcase of various outbreaks, multi-state outbreaks on the CDC website that happened over the past few years that all involve salmonella for the most part. Yes, all salmonella, or few of them have hantavirus or campylobacter that were involved in multiple types of pets. And so that's really important. One of the larger growing populations of pets are backyard flocks. And even during COVID, we really saw an increase in individuals who wanted to have their own food production. And so we see this increase in the growing of backyard poultry. This was an outbreak that is now concluded, although there is a current outbreak going on of salmonella linked to backyard poultry. And so when we and that is, has about 163 illnesses as of today. across 43 states and 34 hospitalizations involved in the one today. That's on the CDC website if you'd like to know more. And so we really want to think about what it is about salmonella that makes it such a good pathogen when it comes to One Health. One of the issues is that the transmission occurs here between humans, animals, and plants, and salmonella can survive very well in those different environments. We also have seen multiple outbreaks occur with poultry, with fruits and nuts, with vine stock vegetables, and salmonella is able to survive in all of these environments. This paper here really showcases how salmonella is able to survive across multiple host species and in the environment, and does that in a much better way compared to E. coli. And what's interesting is that we've seen changes in terms of salmonella, right? We've really seen a decrease in the animal salmonella that we were more familiar with, Salmonella pylori, and it has been replaced with cases of salmonella in humans. So there's a lot that has to do with the way we interact with animals, the way we interact with our environment. And I can show you just some quick data of a paper that we have that goes into these differences. If we inoculated a salmonella cocktail here in different species from different animals, I just want you to take a look at this, and you can see there is such a difference here of the survival of fecal material from different animals across different states, right? And so you can really see those differences here. And if you look at what salmonella survived, each salmonella acted individually, very different across the others. So there's a huge amount of information to dive into salmonella. So in the last few minutes that I have, I want to move away from the bacteria and talk a little bit more about a virus that is also a really wonderful example when it comes to talking about One Health, and that is avian influenza. You may have seen this infographic here because as we talk about RNA viruses, the novel coronavirus changes more slowly compared to influenza virus that we know mutates very well and adapts extremely well to multiple hosts. And you can see that here on the right-hand side. I also want us to keep in mind this growing backyard flock, right? And this is a newspaper article from the New York Times posted recently that has this lovely family embracing their new flock. And we really want to think about how do we educate people and how do we encourage this type of food production and embracing animals, but how do we also get people to think about those collaborative approaches to One Health? And influenza is a great example, as I said, of One Health when we think about how it affects humans as well as animals. There are multiple different strains that can be identified around the world, and we're going to look at just a few of those here. Of course, those mutations occur in both the hemagglutinin and neuraminidase proteins on the outside. There's both larger and smaller recombination events that happen, and those happen here in all of these different hosts. And you may be very familiar with swine flu. You may be less familiar with the role that terns or marine mammals play, but they're all very important reservoirs when it comes to avian influenza. And a couple of examples here. We have adaptation of avian flu, where we have seen this adaptation happen and occur in leading to outbreaks in swine as well as poultry. More recently, H5, H7, H9 infections directly from transmission from poultry, and that's again why we really want to think about how do we communicate to individuals when they have these backyard flocks. In the 2015 AI outbreak, we saw outbreaks in the commercial as well as backyard flocks, right? So I want to point that part out here. So backyard flocks are not necessarily always safe. And this was a huge outbreak because it really resulted in a massive loss of poultry inventory around the country. Another thing to think about is what's happening in our environments in terms of live bird markets and movement of birds. That's been a known issue. And in my university, we are very involved in terms of avian influenza because we have a huge chicken population in the Delmarva area, and it's very important economically, and so we want to make sure that the birds are protected as well as the humans, as well as the environment. And so all of that's really important, not only from a disease standpoint, but also from the economics. And you can see that here on the right-hand side, this is the Delmarva Peninsula. These are all individual family farms, and so it's really wonderful to be able to see people embracing agriculture that way. Again, this is an infographic from the CDC that you may wish to share if you know people who have backyard flocks. We really need to communicate about contact with animals, washing your hands, right, being very careful so that we can reduce transmission when we can. The last couple things I want to talk about is focus back to the environment. When we think about how is avian influenza being transmitted, right, this is a global issue. And all of these, the green, blue, red, black, all of these are flyways from different birds. And you can see they move across continents around the world in some cases. And we have these migratory flyways here in the U.S. But these are each important when it comes to movement of potential diseases as well. There have been some great connections, right, these collaborations in terms of One Health when thinking about ducks that are grazing and where are they grazing in these intensive rice production paddies, right? And so those are really important to think about where we find animals and where we are producing food. Sometimes it's impossible to disconnect the two, but in other cases, we might be able to have an impact. So if we look at what's happening in the United States, we have overlap with the lower latitude migratory wild ducks in terms of where we have a lot of poultry houses. And so we won't be able to move the poultry houses that are there now, but when we're thinking about production of food and industrial animal production, how might we consider the environment and what's happening in terms of the animals? This is a great example of that, showing that viruses were detected in both poultry and wild birds, simultaneously detection, across areas of the world here, right? We have this global connection when it comes to One Health and looking at those flyway patterns. Also, when we think about that global perspective, this is a recent data that comes from the OIE. And you can see on the left-hand side, these are actually outbreaks happening in poultry that were recently identified. And here the red dots are ones that are the outbreaks, the new outbreaks, and then the blue dots are what's called ongoing outbreaks. And if we look at the right-hand side and you kind of match those up, this is where they were detecting those high-pass avian influenza strains in wild birds. So again, there's overlap there. And what can we do to try to understand that, to try to facilitate collaboration among scientists and understanding what's going on in the environment? I want to share this with you, and of course, some of these slides have come from two great colleagues at University of Delaware, Jack Gelb and Chris Williams, and this shows you what some of that effort, that collaborative effort that's ongoing in the United States. Here at the USDA, the USGS, and multiple other agencies in our government have been doing quite a bit of surveillance in the past, really looking for those high-path AI strains so that we can understand what's happening in the environment and help to educate what's happening within poultry production. And that's really important, those collaborations there, including some surveillance. Of course, surveillance in animals has come up when it comes to coronavirus, and so we really want to make sure we continue to have funding and continue to have research that's ongoing for those types of surveillance activities and monitoring what's going on in the environment. So in summary, I hope that we all continue to keep making connections, really thinking about ways to advance food safety and public health. I think it's critical for us to think about how our health is impacted by animals, not only by the pets that we love and embrace and the animals that we use domestically for food production, but also thinking about what happens with those wild animals that are around us that we enjoy if we're bird watching or outside, those wildlife that may be carrying different diseases. How do we interact with them in the environment? As we are, as humans, are really taking over more and more of those spaces, this is becoming even more critical. What can we do as scientists and educators to try to protect those environments? How can we communicate that to individuals, to our students, to consumers, so that they can all understand this? And with that, I thank you very much. And my time is up. So thank you for the opportunity to be here with you.
Thank you, Dr. Neal. Dr. Main, can you please turn your camera on? And you may begin your presentation. Thank you.
Great. So good afternoon, and I did want to thank the first speaker for really setting the stage so nicely for the perspective about One Health, and I know we'll come back to that throughout this panel session today. So welcome to the 2021 FDA Science Forum Food and Chemical Safety session. I'm going to be speaking on behalf of FDA, and the FDA is responsible for 77% of all food consumed in the United States. And we regulate $729 billion worth of domestic food, $124 billion worth of imported food, and over $80 billion worth of cosmetics that are sold across state lines. FDA's regulatory authority over the food supply includes human dietary supplements and botanicals. The employees of CFSAN, CVM, and ORA run the gamut of chemists, toxicologists, microbiologists, physicians, veterinarians, lawyers, nutritional scientists, epidemiologists, social scientists, economists, and others conducting regulatory science-related research. That research is aimed at increasing our understanding of the underlying factors and variables that can contribute to or pose a risk to human safety and health for CFSAN-regulated products. We are a science-based regulatory public health agency, so science is really at the foundation of what we do. You will hear many talks on these areas, so I will give a taste, no pun intended, of some of the innovative science that we are using within CFSAN to address current and emerging public health threats. Next slide, please. So again, just extend the warmest welcome. Let's jump on to the next slide, please. So in terms of innovative science, traditional scientific methods are necessary to stay abreast of current techniques, products, and production. But we live in an ever-changing world, and the consumer drive for novel products, such as cell-cultured foods or edible insects, which include cicadas, by the way, and more information on the production and ingredients of a product requires CFSAN to engage in and encourage the development of new technologies for the detection, reduction, inactivation, or elimination of hazards from FDA-regulated products. Some of the areas where innovation is happening are summarized on this particular slide. So we are using predictive toxicology to inform better decisions while emphasizing our commitment to reduce or eliminate the use of animals in toxicity testing. We do consumer studies, for example, in nutrition and labeling, but also in food safety, to recognize consumer views and understanding in order to provide information that will empower consumers to make well-informed decisions. You're going to hear about some of our work in genomics and metagenomics and bioinformatics to help us clarify the root cause of foodborne outbreaks, support those investigations, and prevent future outbreaks. And on the chemistry side, we continue to develop more rapid analytical methods to set the global standards for measuring chemicals of concern. And that would include chemicals such as the cannabidiols or CBD, the PFAS substances, the per and polyfluoroalkyl substances, metals, and we also do risk assessments to inform policy decisions aimed at reducing consumer exposure to potential hazards in the products that we regulate, including food. So I'd like to give you just a brief introduction to each of these as just a few examples about some of the innovative science that we are engaged in CFSAN. Next slide, please.
Dr. May, can you please check your camera to see that if it's on? Thank you.
Let's get back. It was on. It got me kicked out. Let me get
dropped out
back.
Momentarily. Here you come.
I'm coming back in, Chad.
Great, thank you. There we go. Thank you.
There we go. There we go. Next slide, please. Okay, on to predictive toxicology. Some examples about predictive toxicology and the importance of it I'll highlight here. First, predictive toxicology can be used as an alternative to traditional in vitro or in vivo methods and animal testing, and CFSAN has been strongly engaged in this area. Currently, we are publishing multiple papers on the use of a predictive toxicology tool called the expanded decision tree. The expanded decision tree classifies compounds according to their chronic oral toxicity using a sequence of structure-based questions to which the answer either refers the user to another question or assigns the substance to one of 6 classes of toxic potential. The combined expanded decision tree and threshold of toxicological concern approach is suitable for screening, prioritization, and safety evaluation of substances with low exposure scenarios, along with the safety evaluation of mixtures. This is so important given that much of CIFSAN's work is in the post-market setting, although the expanded decision tree can also help with pre-market reviews. Another exciting area is developing alternative methods to animal testing for cosmetics. Look for 3 posters describing the development of new methods and the establishment of already validated methods to identify potential skin sensitizers. The new alternative methods combine predictive computer modeling and in vitro test methods to minimize animal testing. Legislation has considered banning the use of animals for testing cosmetic ingredients, so progress in this area is critically needed. And so please look for posters on both of these topics. Next slide, please. On to consumer studies. CIFSAN has a well-developed program in consumer studies providing robust research to understand consumer thoughts and views so that we can provide them understandable information, empowering consumers to make informed decisions. For example, we use focus groups to understand consumer attitudes and to determine the most effective venues, formats, and content to ensure our communications are accessible and understandable for consumers. Another application is through experimentation of how consumers understand claims and how this information can result in erroneous beliefs or confusion. These experiments provide empirical data to help support agency actions on product labeling. A third aspect of surveys is focusing in on consumers' knowledge and beliefs that offers important information to policymakers, educators, and communicators about the need for targeted public information to address knowledge gaps and better inform consumer decisions. Look for Amy Lando's presentation on the results from the 2019 Food Safety and Nutrition Survey. There are also posters on a child obesity focus group study and on data from a raw flour analysis that we did showing that most consumers are unaware of the risk from consuming raw or uncooked flour. Next slide, please. Switching into genomics, metagenomics, and bioinformatics, we've been using tools like DNA barcoding, which has been utilized to identify sequence data specific to an organism distinct from another species. For example, we have used DNA barcoding to identify seafood species. And we are taking this technique further by applying it to dietary supplements and botanicals. This will be used to test the authenticity of labeling and to prevent economic adulteration of these products. Another area of genomics, metagenomics, and bioinformatics that we are well known for is Genome Tracker. The Genome Tracker network is the first distributed network of laboratories to utilize whole genome sequencing for pathogen identification. It consists of public health and university laboratories that collect and share genomic and geographic data from foodborne pathogens. The data, which are housed in public databases at the National Center for Biotechnology Information, or NCBI, that data can be accessed by researchers and public health officials for real-time comparison and analysis that can be used to speed foodborne illness outbreak investigations and reduce foodborne illnesses and deaths. The human and economic impact of predictive identification or rapid identification of disease and thus outbreak prevention or disruption is tremendous. And you can see posters that can be found on both the Genome Tracker network updates and the economic impact as well. Next slide, please. So talking more about methods, an important role that we have is to help to develop rapid analytical methods. And as I just mentioned with regard to Genome Tracker and whole genome sequencing, methods that are robust, accurate, efficient, and rapid can save lives and can help improve public health. And we are currently focusing on a variety of such analytical methods. With the increased marketing of cannabinoids and hemp products, we needed to produce methods to analyze CBDs, toxic elements, pesticides, and microbial contamination in a variety of products, including beverages, food, and pet products, and powders and oils. We are currently sampling over 200 products, and we plan to sample up to 3,000 products long-term. Another area of concern is the mixture of e-waste or electronic waste in the recycling stream, which can contaminate food contact materials, for example, plastic being recycled for food packaging. The primary concern is that heavy metals, flame retardants, and polymer degradants that are not authorized for food contact could make their way into recycled materials that are being used for food contact. Concerns of misidentification when utilizing our current methods pushed us to develop and test a rapid screening method we are currently using for a survey of marketplace samples. Next, there's great interest in metals in food, such as arsenic in infant rice cereal and apple juice, and we have continued creating methods that are rapid, specialized, and lower cost. Cost is important both for our own testing as well as to incentivize greater testing of foods by industry. For example, we have adapted an off-the-shelf well water testing kit for testing infant rice cereal. By producing validated methods, field investigators can test products quicker and more efficiently. Another new area is that of portable screening devices. Consumers now have access to marketed devices that use smartphone apps or other technology that are easy to use and user-friendly. However, these tend to have low functionality and spectral range and need testing. We plan to develop screening methods that employ such portable technology. Look for the poster on the development of non-target screening methods based on vibrational spectroscopy instrumentation and chemometrics for applications such as milk powder authentication, seafood decomposition analysis, and determination of plasticizers in tubing. Next slide, please. As I mentioned earlier, CIFSAN is also responsible for cosmetics and cosmetic safety. And I would encourage you to look for several posters that we have available on cosmetic safety. One poster provides an overview of new methods developed to determine the mercury levels in certifiable color additives. CIFSAN certifies color additives, and these are additives that are used to color foods, drugs, cosmetics, medical devices, and in some cases, the human body. The FDA pre-approves color additives, and mercury is a known impurity. Next slide, please. Hopefully, as you can see, CIFSAN continues to test industry and consumer technology while developing our own to meet consumer demands, create a safe product marketplace, limit animal testing, and provide information to empower consumers. We look forward to continuing to serve you and the country, bringing our best science to food and cosmetic safety. Thank you for joining us, and enjoy the session.
Thank you, Dr. May. Dr. Rice, you can turn your camera on. And you can begin your presentation. Thank you.
Well, hello, everyone. I'm honored and thrilled to be part of this session of the FDA Science Forum and speaking on a panel of such excellent scientists. Today, I'm going to be presenting on FDA support of recent foodborne illness outbreak investigations, and I think what I'll be presenting today ties in really well with Drs. Neal and May's presentations earlier on both on One Health and some of the technological advancements we've made to help support outbreak investigations. There are a lot of players in the prevention, detection, and response of foodborne illness. It's very much a team approach spanning several agencies at the federal and state and local levels. And so in my brief presentation today, I'll be highlighting the FDA contributions of recent investigation efforts and also emphasizing how these other partners are so important in the investigations that we participate in. So, as I said, it's a collaborative approach. FDA is a partner in response to outbreak of foodborne illness for most human and animal food products, the products that we regulate. And it's a collective effort spanning the Office of Food Policy and Response, the Centers for Food Safety and Applied Nutrition, where Dr. Main is director, the Center for Veterinary Medicine, FDA CORE, which is Coordinated Outbreak Response and Evaluation, the Office of Regulatory Affairs, where I work, compliance programs, inspection programs, and laboratories. And actually, I'm a laboratory person. I direct the Office of Human Animal Food Laboratory Operations for ORA, Office of Regulatory Affairs. A little bit about FDA's Coordinated Outbreak Response Evaluation Group. They manage response, surveillance, and post-response activities, and they're a group that we, in my shop with laboratory operations, we interact with staff and core pretty much on a daily basis. In the Office of Regulatory Affairs, we have inspectional laboratory testing support. So there's the Office of Human Animal Food Operations that oversees domestic food that's in commerce in the USA. And they have staff who perform inspections of food manufacturing facilities, food distribution centers. They are the staff who collect samples that come to the laboratories that I manage. They also perform trace back if there's an outbreak and we need to trace some implicated product back. And then manage the logistics of recalls, which are really sometimes quite complex. And then there's the Office of Regulatory Science, where I'm based. And we provide laboratory support for surveillance sample testing, outbreak sample testing, results communication, and coordination with the FDA centers like CDN and CIFSAN and CORE. There are counterpart scientists, peers and colleagues in laboratories within the centers that we work with on very closely for not only outbreak investigations, but also technological advancements and method development and so on. So the nuts and bolts of an outbreak investigation. I'd like to emphasize that I can't say more about our state partners and how important they are in the foodborne illness outbreak investigations that we perform. They're usually, FDA gets involved when it's a multi-state outbreak investigation. We always end up collaborating and coordinating with our state partners and the CDC. But it's a coordinated effort that not only to address outbreaks as they occur, but to help mitigate the impact of these outbreaks and to prevent outbreaks from ever happening. There's epidemiologists that are helping with the investigation. There's inspections. There's sample trace back. There's implicated product distribution that needs to be traced. And then, as I mentioned, managing the product recalls. The goal is early identification of illness clusters, and then rapid and effective regulatory response to remove contaminated food from distribution, and then outreach to inform consumers. And so although foodborne illness remains a significant overall burden to human illness, there is good news that technology and enhanced response capabilities have reduced the size of most of the outbreaks that we're working with now, as in fewer cases of human illness. And in general, the outbreaks are detected sooner than they were in years past. And that's largely due to some of the genomics and metagenomics technologies that Dr. Maiden mentioned previously. A little bit about FDA CORE. So it was established in 2011, and it brought together expertise in medicine, public health, epidemiology, and science to coordinate efforts to identify, stop, prevent foodborne illness outbreaks. And so when an outbreak is associated with an FDA-regulated product, there is a response team stood up in CORE that coordinate FDA's response efforts. And as I said, in any given day, there's usually multiple outbreaks that we're working on, and staff in my laboratory organization interact with staff in CORE on a daily basis, and it really is a productive collaboration. And to give you a sense of CORE's contributions since they were stood up in 2011, and these numbers grew almost a daily basis, but been working on up to 1,000 potential outbreaks, responded to over 200 outbreaks that potentially linked to FDA-regulated products. They identified contaminated food products in over 100 outbreaks, and warned consumers to avoid implicated products through more than 400 public notifications. Over 251 food recalls are initiated, and multiple targeted assignments, including for food facilities, farm investigations and inspections, record collection, and sample collections related to these outbreaks were initiated by CORE. But as I said, the C in CORE is coordinated, and so it's a coordinated and collaboration among multiple partners, including within FDA and external to FDA. So this is a list of some of the primary foodborne pathogens that we work with. There are certainly other organisms that we encounter, but these are the main ones. And to link this back to Dr. Neal's presentation on One Health, most foodborne pathogens are zoonotic pathogens, and so the interventions that we take largely take a One Health approach. And I'm certainly an advocate of One Health to addressing the burden of illness in both humans and animals. And so E. coli 157H7 is an important one that we work with. Most of the outbreaks of late have been linked to leafy green, primarily lettuce. Salmonella enterica, Listeria monocytogenes, and then the protozoan parasite Cyclospora cadenensis, and some viruses, hepatitis A and norovirus, which we've had a number of outbreaks linked to those in recent times. So some recent investigations, FDA has provided technical and epidemiological and inspectional laboratory support for several recent outbreaks of foodborne illness. I'll start with E. coli 157H7 and leafy greens. But I mentioned Cyclospora in fresh produce. That's primarily imported produce that we see that. Salmonella enterica in a variety of products, and we're working on an outbreak right now in a cashew brie product. And then hepatitis norovirus in fresh berries and produce. Again, those are usually in imported products. So on the E. coli 157H7 front, we've had ongoing investigations in the lettuce growing regions of the U.S. In 2020, there were 2 outbreaks that were multi-state of E. coli 157H7, and epidemiology and traceback determined that they were linked to leafy greens. And when we looked at the whole genome sequence analysis, the genetic fingerprinting of these isolates, we could see that they aligned with the cases that we were seeing in 2020, but they were also associated with prior outbreaks in 2018 and 2019. And so because of this reoccurring incidence of this particular strain of E. coli in leafy greens, primarily lettuce, the FDA has initiated several sampling assignments that were focused on the growing regions in Arizona and California, looking at both environmental samples and product samples for this pathogen and others, including Salmonella, and there's other hemorrhagic E. coli of interest as well. And in fact, there's an assignment going on right now, a surveillance assignment, which is looking hopefully to prevent occurrence of illness by detecting it before the product gets to market in the Sal Bowl, California, targeted towards leafy greens and lettuce. Another recent outbreak investigation late last year and into early this year involved Salmonella poxstem illnesses. There were 7 cases in 4 states. We were notified by CDC, and then we stood up CORE to help coordinate the investigation. The epidemiology implicated smoked fish. Pretty quickly, FDA was able to identify a firm in Brooklyn, New York that manufactured the product, and it was linked back to tuna that was imported to the U.S. from Indonesia. So FDA collected 15 product samples and 1 environmental sample and submitted them to one of the labs in my shop. And at the time, put the Indonesian firm on increased screening. And after testing the product, we found that 4 of the samples collected had Salmonella in them, and the positive samples and products were from the Indonesian supplier, and the isolates aligned with the outbreak strains that were being seen. And so the FDA has some tools called import alert and detention without physical examination that we use for imported food products, and they're excellent tools that allow us to prevent contaminated food products from entering U.S. commerce. And so this particular firm was put on an import alert to keep tuna from this supplier out of the U.S. until they could demonstrate that their product was salmonella-free. Another recent investigation was on Listeria monocytogenes in Spanish-style fresh and soft cheeses. Again, it was a multi-state outbreak investigation. CDC had the initial signals and stood up a core. The FDA did an investigation. Samples were collected and tested. We worked closely with the state lab that was initially involved in this. And Listeria monocytogenes was isolated from product. The product was recalled, and the cases have since stopped, but whole genome sequence analysis was used to determine that the product found in the cheese samples aligned with what was being seen in the humans. And I'd like to emphasize the importance of Listeria monocytogenes, especially in ready-to-eat products. It's one of the least common foodborne pathogens, but it has a very high case fatality rate. So it's really a high priority organism for us to detect and then remove contaminated product. And so on the radar, you know, on any given day, we're looking at signals to see what's going on and if outbreaks are occurring, and FDA and CDC are continuously tracking clusters of foodborne illness, and then we apply epidemiological tools to detect outbreaks. So currently tracking a few potential outbreaks without a known food source yet. I mentioned there's one active outbreak in cashew brie where product has been recalled. It was linked to salmonella. And so, you know, collectively, FDA, state and local food protection agencies perform risk-based inspections and food sample collections. And a lot of these efforts are preventing outbreaks that we never even know about. And then we have the tools and resources in place to respond to outbreaks as they occur. And with that, that is the end of my presentation.
Thank you, Dr. Rice. Dr. Edwards, can you please turn your camera on? And you may begin your presentation once your slides load. Thank you.
There we go. All right, good afternoon, everyone. And thank you to the organizers for this invitation to be part of the session on innovation in food and cosmetic safety. So as you will soon see, what I'm going to talk about today really builds upon what other presenters have already discussed. When we talk about One Health, we know that livestock, poultry, and pets, all ill and mental health of people. Working together on One Health does help across the human, animal, and environment to maintain the healthy ecosystem that involves us all. Seeing this beginning slide, how many with pets at home have wondered the question posed here? It's really astounding the number of different ingredients that appear in pet foods today and or things that pets may ask us for around the house. In the division of animal feeds, we see pre-market as well as post-market reviews for ingredients that go into animal diets, not only of pets, but those of livestock, poultry, and aquaculture species besides. The submissions become more and more complex every year, and yet we have to keep up with the innovations in order to ensure the safety of human and animal health. Although a few years old now, the data on this slide indicate the impact of those innovative ingredients and the mixing of animal diets in the U.S. The dots on the map indicate animal food manufacturing facilities. From the east to the west across the country, animals are consuming almost $300 billion worth of animal food each year. As a reminder, different from humans, the domestic animal species tend to consume very similar diets for much of their lives. This is one of the big considerations in assessing the safety of animal food ingredients. It is also important to consider that a food an animal consumes today may become the meat, milk, or eggs are on our own plate tomorrow. Animal food is manufactured in every state in the United States and includes those foods that are made for all different types of species. It continues to grow and makes a big impact both on our mental as well as our physical health. Now here's a quick quiz. You thought you probably wouldn't get a quiz today, but some of the animal food ingredients that we see may sound foreign from what we see on our human food labels. In reality, though, some of the terms have been around for quite some time. So here's the quiz to remember and ponder as I go through this talk. Does anyone know the definition of bagasse, corn gluten meal, or de-oiled corn distillers dried grains with solubles? All of these are animal food ingredients used on a daily basis. And so stay tuned for answers to this quiz later in the talk. You don't have to come up with the answers all on your own. I think many of you are familiar with the Code of Federal Regulations, as well as the Food, Drug, and Cosmetic Act. Obviously, in the Act, food is defined as articles used for food or drink for man or other animals. Any substance that's intentionally added to an animal food must either be an approved food additive listed in 21 CFR 573, 579, or a substance that's generally recognized as safe, GRAS, for an intended use. In the animal food space, in coordination with state feed control officials, which we've heard of the coordination in other talks today, CVM also recognizes ingredients in the AFCO official publication as being acceptable for use in animal food. Now let's talk a little bit about some of the innovative ingredients and some of those ingredients that have been utilized in animal foods for a long time. Byproducts of the production of human food have been used for centuries to feed animals. Examples appear on the slide, things such as the wheat middlings that come from processed wheat for flour, those peels, rinds, and pulp that might be left over after the processing of fruit and vegetables. And now today, recycled foods or foods that maybe past their sell-by date but still be safe, things such as chips, cookies, candies, or bakery products. And they end up not going to the human food supply due to quality characteristics, but yet they may still be safe. These human food byproducts can be either in a typical commerce stream or can be a one-off stream where you don't see those all the time. Today's animal will include these byproducts, but they may be processed ways or have quality defects that do not impact safety, as I mentioned. There are several initiatives underway, both in the public as well as the private realm, to lessen the food that is thrown away in a landfill. And one of the options that is mentioned often is that food being utilized in new ways as animal food. Obviously, these different products still need a safety determination to ensure that the animal food is safe and any resultant human food from those animals. We have seen many new products and different fractions of products over the last few years. Another place that we have been looking at the promotion of innovation and emerging technologies and product safety are things that you see on this slide. Most simple animal food ingredients have already been approved in one way. I think that those would be called the low-hanging fruit. The submissions that come in today for us to review are complex and have things such as genetically engineered organisms producing specific thermostable enzymes come in today, not such low-hanging fruit anymore. As livestock producers move away from antimicrobial drugs administered in feed, they'll be looking to provide optimal animal nutrition and support gut health with things such as enzymes, probiotics, and prebiotics. This is a prevention approach versus a treatment approach, and the healthy animals through diet rather than through medication. So we've been successful in moving towards this judicious use of antimicrobial drugs. We've also created a pressure point where we're going to see an increase in novel and complex animal food ingredients. Adding to the pressures that on animal food ingredients are things such as consumer preferences. These consumer preferences may indicate where they're asking for certain ingredients to be included in animal diets, or on the flip side of the coin, for the absence of certain ingredients within animal diets. CVM will see more food ingredients coming in for pre-market review with these new additional ingredients. CVM reviewers do receive more data to dig through to adequately assess these new and complex ingredients. for both the animals eating the food ingredient and the humans eating the edible tissues of those food-producing ingredients or food-producing animals, it's important to look at the safety. The couple pictures that are shown here on the slide are two new ingredients that have come through for animal food in the recent years. The top picture is that of black soldier fly larvae. This is a source of protein and oil for things such as poultry, salmon, and maybe soon other animals. These insects are raised on food scraps, which would otherwise have gone to waste. Instead, the insects eat the food scraps and are turned into high-quality food for other animals. CVM looked at the fatty acid profile to see if it matched those needed by chickens and fish, and thus determined safety and usefulness of the ingredient. The lower picture on the slide would be that representing marine microalgae. This would be single-cell algae farmed to produce DHA for dogs, thus saving pet owners from having to get it from fish taken from the ocean. CVM also looked at safety for those dogs and whether the DHA would be useful in the dog's diet. A couple examples of ingredients that have come through CVM in the past several years. While looking at these different ingredients, obviously safety is an important part of the review. The safety of an ingredient does differ amongst species, though. Our safety reviewers in my division really do like to remind me that a dog is not a cat, is not a rat. When we consider safety, we do so across many species, including the safety of any human food that the animal may be producing. Earlier, I talked about human food byproducts being a part of the animal diet. Additional considerations for safety need to be taken into account due to these species differences. A couple examples here of some human foods and minerals that have been found harmful in certain animal diets. You might not expect this across the different species, but with those differences across the different species, you see things such as xylitol, which appears in many human foods but is harmful to dogs. Things such as sulfur amounts to harmful to cattle, and copper can be harmful to sheep. Safety is key. What an animal eats today may be the food you eat tomorrow. As we look into the crystal ball in terms of what things might be next in line for animal food ingredients, that innovation really is taking many different shapes. Some of those trends are easier to predict for others, but here are a few that I'll list as we're looking to the future. You may have heard of cell-based animal foods mentioned in an earlier talk today, things such as hamburgers grown in cell culture, but the same is also happening in pet food. One innovation that we have seen is to grow mouse cells up to be part of cat foods. It's also occurred where growing other traditional livestock animals in a cell-based culture for other types of pet foods, for dog foods and for cat foods. So one of the interesting new innovations. We've all heard a lot about hemp and hemp seed ingredients. These two are occurring within the animal food space, both the hemp seed oil as well as what's left over after pulling off the oil, the hemp meal, are things that we'll see. We've heard of shareables. Now, shareables is not necessarily a new ingredient per se, but one in which an ingredient or a food may be something that both the human and its pet could eat, where you could share that with your pet. We're not so sure about the safety of these and haven't had to look at those yet, but that is something that may be coming, where we're evaluating not only safety to the animal, but also safety to the human, because they're both eating it together. We have seen innovations where pet foods would be intended to be reheated at serving. So you not only would heat the food for yourself at dinner time, but you would heat the pet food, hopefully not on the same plates, and hopefully being safe between those. But that is something that has come, and it's part of the safety consideration if you're looking to make sure that you keep safe for both the pet as well as the human who's handling those foods. And I think I talked a little bit about it, but taking today's food waste and feeding it to livestock is something that's been done for a while. But new components and new human food leftovers are things that are making it into the pet food and livestock food supply. Some examples of this is where human food leftovers would be mixed together and enzymatically digested to create part of an animal's diet. This would help with furthering the reduction of food waste here, and it is something that lots of firms have been talking about, and we've been thinking about how to do the safety recognition of that. So now, does anybody remember the quiz from the beginning? If you have looked up the answers on Google in the interim, or if you know the answers and we're thinking about them, here's your quiz answers for today. So bagasse. Bagasse is that portion of the stalk of sugarcane after removal of leaves and tops that remains after extraction of the juice. Bagasse can be used as a source that has a lot of fiber in it and is utilized in different animal diets. Did anybody know what corn gluten meal was? Well, it's the dried residue from corn after the removal of the part of the starch in the germ. Separation of the bran by the process employed in the wet milling manufacture of cornstarch or syrup, or by enzymatic treatment of the endosperm. And it doesn't have any wheat gluten in it. So when you see corn gluten meal in your animal diet, it's not quite the same gluten as you would see in wheat gluten. And the last one, the de-oiled corn distillers dried grains with solubles. Now that's quite a mouthful, but it's a portion of the corn that is resulting from the solvent extraction of oil from corn distillers dried grains with solubles, which come from ethanol production to result in a crude fat content of less than 3% on an as-fed basis. And it's a source of protein that we see in many livestock diets. So today, hopefully you have found out a little bit about some of the animal food ingredients, learned a little bit about some of the innovations that are coming down the line, and remember that One Health, talking about the health of animals and humans and the environment, are all tied together through the safety that is looked at. I appreciate the opportunity to be a part of this panel. And for further information, you can visit the website on this slide. So thank you very much.
Thank you, Dr. Edwards. Dr. Camacho, can you please turn your camera on? And you may begin your presentation. Thank you.
Thank you. So good afternoon. Thank you, Shed, and good afternoon, everyone. I'd like to thank the organizers for the opportunity to showcase some of the activities that we are conducting at the NCTR to contribute data to evaluate cosmetic safety. So what I'd like to do first is to give you a very brief overview of our centers, the National Center for Toxicological Research, and then showcase a couple of examples of studies that we've conducted to provide safety data on different products, cosmetic products and ingredients, in this case tattoo and permanent makeup ink, as well as aloe vera leaf extract. And then a new study that we started a couple of months ago that is evaluating a novel tool that may be of use for in vitro permeation testing, for instance, of cosmetic ingredients. So NCTR, or the National Center for Toxicological Research, was funded 50 years ago, almost to the date, and it is a non-regulatory research center of the FDA. So unlike the other centers, we are located in Arkansas, south of Little Rock, and we share the campus with the Office of Regulatory Affairs, Arkansas Laboratories. So the campus is quite wide. It includes about 30 buildings, and we have a staff of 550 plus members. And these include full-time researchers, staff fellows, and postdoctoral fellows, as well as a number of on-site contractors, including animal care and pathology contractors. So the NCTR mission is twofold. So we conduct scientific research to generate data for decision-making by the FDA product centers, as well as we develop and support innovative tools and approaches that the FDA can use to protect and promote individual and public health. And so in this sense, NCTR supports all offices and the product centers at the FDA to support their regulatory mission and decision-making. So we have quite a broad scientific expertise, and I hope you had the opportunity to listen to some of the talks of my colleagues over the yesterday and today on AI, bioimaging, microbiome studies. Here I'm just listing some of the studies more focused on that we've conducted over the years, more focused on the evaluation of cosmetic safety. And these include rodent toxicological studies, including chronic dermal studies, both in vivo and in vitro skin permeation studies, pharmacokinetic studies, biodistribution studies, as well as a wide variety of genotoxicity and mutagenicity studies. And we also conduct and have expertise on the chemical and microbiological analysis of cosmetic products and ingredients, for instance, to characterize their heavy metal load and potentially identify pathogenic agents. And so what I'll like to do first is, and here I'm just showing some of the chemicals that we've evaluated over the years on ingredients that are widely used in skincare products, for instance, alpha and beta hydroxy acids, aloe vera, ricinyl palmitate, more recently CBD or cannabidiol, as well as permanent makeup pigments and tattoo inks. And many of these studies, in particular the genotoxicity and the bioassays, have been conducted often under the sponsorship of the National Toxicology Program through an interagency agreement between NCTR and NIHS DNCP. So the first study that I'd like to share with you today is an ongoing microbiological survey of tattoo inks and permanent makeup inks. And this study is being led by my colleague Seong-Jae Kim at the Division of Microbiology with the support of the Steve Sun's Office of Cosmetic and Colors. And this study was really initiated because about 10 years ago, there were several outbreaks of tattoo-associated skin infections with pathogenic bacteria. And so this really raised awareness and recognition that tattoo inks can be a potential source of bacterial infection. And so what we've been conducting at NCTR for a few years now is a survey of unopened tattoo and permanent makeup inks that are sold in the US and conduct an evaluation of the microbial contamination, including of pathogenic bacteria. And there's been a couple of reports published already, so I would encourage you to look for them if you are interested to learn more. Just to give you an idea of the findings, here I'm showing the geographical distribution of the manufacturers from which products were evaluated this far, and this includes a wide range of manufacturers both in the US as well as imported products. And you can see that there was indeed widespread microbial contamination of the product. So in red, I'm showing the number of the products that were found to be microbiologically contaminated versus the total number of products. And you can see that the red really stretches around the map. When looking for the identity of these bacteria and as well as fungi were identified, most of them fall on the genera, species from the Bacillus genera, and about half were potentially clinically relevant isolates. However, no mycobacteria were detected on the almost 200 products evaluated this far, but there were some recalls initiated by Steve Sun based on these data. So switching gears, I'd like to share also another study which we conducted to assess the photocarcinogenesis of aloe vera leaf extract in hairless mice. And this study was conducted and led by my colleague, Dr. Mary Boudreau at the Division of Biochemical Toxicology, and it had the sponsorship of the National Toxicology Program. And all the study report was published in the NTP Technical Report Series, and it's product report 553 in case you would like to learn more about this study. So as you are probably most aware, aloe vera leaf extracts are widely used as a cosmetic ingredient in a wide range of cosmetics and skincare products, ranging from lotion, shampoos, and wipes. However, there was uncertainty on whether aloe vera in skincare products could enhance the induction of skin cancer by UV radiation. And so we conducted a one-year photocarcinogenesis study to evaluate whether the topical application of various aloe leaf extracts could alter the effects of stimulated solar light on the skin of hairless mice. And we tested different extracts, including the whole leaf extract, the decolorized whole leaf extract, as well as aloe gel. In the next couple of slides, I'll show you some graphs with the study findings. I'm going to show the female data on the left and the male data on the right. And here it's listed the neoplastic skin lesions that were observed in the rats, both individually and all combined. And in this slide, I'm showing the controls of the study. So as expected, the simulated solar light induced an increase of both the incidence and multiplicity of squamous cell skin neoplasms in the exposed rats. And this effect was dose dependent. So the longer the exposure to the light, the higher the incidence of the skin neoplasms. Also as hoped, the control vehicle cell had no effect. So here in the black, I'm showing the control cream plus the simulated solar light, or just the solar light, simulated solar light in white, and you see there's really no difference between the two groups. When the cream was formulated with aloe vera leaf extract, we observed weak enhancing of the effect of the photocarcinogenetic activity of the simulated solar light. And so again, just to illustrate the data here, and in this graph I'm showing the multiplicity of squamous cell skin neoplasms in the aloe gel groups, and you can see in the females, compared to the simulated solar light only group, we did have an slight increase of the neoplastic skin lesions. And this was observed in females exposed to aloe gel group, but not in the males in this case. And so the conclusion is that aloe leaf extract, when applied topically, can have a weak enhancing effect of the simulated solar light neoplastic activity in the skin. The last study that I'd like to share today with you is a study that is being conducted with the support of the FDA chief scientist and in collaboration with the colleagues from the Center for Food Safety and Applied Nutrition, the Center for Drugs, and Center for Veterinary Medicine at the FDA, as well as colleagues from the NIH, NCAT, and the NISTM. And what we are evaluating here is the potential of 3D bioprinted human skin to be used in vitro permeation testing. So as we know, it is important to understand whether a compound that is applied to the skin, such as a cosmetic, remains on the skin, or by the contrary is absorbed through the skin to reach the bloodstream and potentially reach distal target organs. And so the assessment of rate and the amount of dermal penetration of cosmetic ingredients is a key consideration in their safety profile. In terms, both in vivo and in vitro testing can be performed, and in terms of the in vitro absorption studies, excised human skin is the current gold standard model. However, there's ongoing research efforts to develop and validate non-animal but human-relevant alternative models of native human skin. And so in this project, what we are evaluating is the potential of a 3D bioprinted human skin model that has been developed at the NIH NCAT in the laboratory of Dr. Mark Ferrer for in vitro permeation testing. And so NCAT is manufacturing the bioprinted human skin, shipping to NCTR, where we are conducting further characterization of the model, as well as in vitro permeation testing. And here I'm showing you can see the front cells, which are the setup we are using for the in vitro permeation testing, with the inserts with the Bio-British Skin Mountain. And so this is a new project, and hopefully in the upcoming months we'll have exciting data to share with everyone. And so this is where I will stop. I hope I was able to illustrate some of the efforts that we are conducting at the NCPR to develop and evaluate novel tools that can be used for cosmetic safety evaluations, as well as some studies that we have conducted to address regulatory data gaps. And thank you for your attention, and I'll be happy to take any questions. Thank you.
Thank you, Dr. Camacho. Excellent presentations from all of our speakers. I would now like to invite everyone to turn on their cameras, and we'll now turn it over to Dr. Jeff Ward to begin the panel discussion. Thank you.
Okay, thank you, everybody. Just waiting for Dr. Rice to join. There we go. Okay. I just told Chad that I was happy that we had more time than was allotted for this discussion. I think this could be the most exciting part. Thank you, everybody, for your very good, excellent talks. I think the people from the FDA always sort of relish these opportunities to reach out and talk to the general public and whoever else. And I know that we always relish reaching out and interacting with people like Dr. Neal to get their perspective on what we're doing as far as the science side of things go. So we have come up with a number of questions, which for full disclosure, we did share with our panel, just so there weren't any communication issues, so everybody knew what we were going to be discussing. So we tried to have some general questions that everybody could get involved with, although a couple of them are a little bit more focused than others. So to kick things off for the first question, and this is really sort of a One Health-oriented question for all the speakers. I think everybody could have input on this. We have all seen the impacts of diseases that affect many species, humans and animals. Dr. Neal and others have talked about COVID-19 and the issues involved there, but there are things like Ebola, which we know has an animal reservoir, obviously things like BSE, mad cow disease, and even diseases like African swine fever virus, which doesn't affect humans directly, but it does get into wild populations. It has economic impacts, and it also has pretty big nutritional impacts because a lot of parts of the world depend on pork for their high-quality protein requirements. So considering all those different zoonotic or other diseases that affect humans in one way or another, what else do you think that we can do to increase our ability to detect these pathogens or to prevent their spread in FDA-regulated products? And I'll let anybody who wants to jump in on that.
So maybe I'll just start us off a little bit. So what first comes to mind is, and I think many of us are having these conversations about the silver linings from the pandemic, right? What have we learned that we can take away, that we can really continue to invest in? And to me, one of those is wastewater-based epidemiological approaches to surveillance. And the FDA is very much involved in this and sequencing wastewater and looking at COVID analysis that way. The Centers for Disease Control and Prevention have launched a national wastewater surveillance system. And I think capitalizing on what academics and private-public partnerships are doing around the world to look for SARS-CoV-2 and using really increased diagnostics like digital droplet PCR, which allows us to have better precise detection, we can use that to monitor for almost all of the agents that you just discussed, the viruses, bacteria, certainly, probably a little bit difficult with some of the prions. We might have to figure out what we look for there, in particular certain proteins. But I really think that this infrastructure that's been developed literally around the world, and it's so exciting to see it start and populate and scientists really get involved. And what's really neat is even in the state of Delaware, we're using it with students and in middle schools and high schools, and I think we've got some people listening to us from those schools now. And these students are getting involved and they're learning about detection of these agents, from their own wastewater and around the surface water and everything else. And so I'd love to see us be able to continue to use those type of surveillance mechanisms to really continue to look for diseases before we see them in our populations.
And I guess I can go next from the FDA perspective. And I agree with what Dr. Kniel highlighted. And a lot of it's that public health infrastructure. And we have to invest in the public health infrastructure. We never know what's going to be right around the corner. But having labs that are ready to go, that are trained, that have sequencers available, that understand the bioinformatic platforms. One of the advantages of our genome tracker network is that we have these labs around the states and also in other countries. They know how to share the data. They know how to upload the data into NCBI so people can access the data around the globe in real time. And it's taking advantage of those kinds of networks and experiences that are so critically important. I do agree about the wastewater issue as well. We have been using wastewater surveillance as part of our foodborne pathogen work. So we've tested wastewater for things like norovirus and Hep A and cyclospora as well. And so that is work that we have developed, including developing analytical methodologies to pull out things like cyclospora out of wastewater. And now we are utilizing those same skills from genomic sequencing, from wastewater surveillance, and we are doing some work with COVID variant detection as well. So it's taking these investments we've made in the public health infrastructure, making sure those labs are current and functioning and know how to do this work, and being able to deploy them as we end up with any future public health challenges like we've had with SARS-CoV-2.
Yeah, and I'd like to jump in. This is Dan Rice. I really appreciate the comments by Drs. Kniel and Main. And I agree, a robust global public health laboratory network is going to be key with the One Health approach. I'd also like to highlight technology in our laboratories as well. And so 10 years ago, we were all really excited about whole genome sequencing. It's pretty routine in our labs now and has been just a really fantastic tool. The next thing to come is going to be metagenomics, and what I think I'm looking forward to is the day that we don't have to culture confirm and isolate to take regulatory action, that we have actionable data based on the sequence data. And metagenomics is promising to move us in that direction.
If I can just jump in, I just wanted to raise awareness that we are actually at NCTR conducting a study on looking at the wastewater SARS-CoV-2 detection. And we've collected samples for over 1 year now. So we started really early on since April last year, and we've been collecting samples every 2 weeks. And actually, we have a poster. So if you are interested to learn more about it, I would really encourage you to look for the poster. And it has been a very interesting study. We really followed the time profile of the cases, and we are working with the Arkansas Department of Health to try to relate to reported clinical cases. And yeah, it just shows how powerful that technique of community level surveillance really can be, not just for COVID-19, but to other pathogens in the community.
And one of the things I'd like to mention too is the things that we've learned over this past year about biosecurity on the human scale, whether it's social distancing, vaccination, or the wearing of protective coverings. Those are things that are good to be a part of whatever biosecurity it is, whatever disease it is. Well, we see in modeling for things like African swine fever, where the spread of African swine fever may be within feed, but it may be attributed to feed, but it may actually be on feed trucks, or it may be through feed totes, or things that are associated with the feed. And I think it's likely that something like that probably comes in on a person, and then maybe spread might be through machines and that kind of stuff. But biosecurity is something that is not only applicable to the animal space, but it's also applicable in the human space that we've all experienced over this last year.
Great. And as sort of a follow-up to that question, and everybody on the panel knows that the FDA isn't the only player here. There's the USDA, there's the EPA, there's even the Department of the Interior and the Fish and Wildlife Service, and there are a lot of different players involved here. Are there any particular issues involved on that side as far as interaction, communication between parts of the government that could be improved, where you see that there are holes that need to be filled? And maybe the answer is no. I don't know. But it's really more of a global sort of a One Health approach to it.
I think with communication, the answer is always yes, there's room for improvement. I don't have any personal examples, but I know that, you know, we struggle with really timely and effective communication, and we have for years, and we keep trying to improve that, but there's always room for improvement for collaboration and bringing down the silos and communicating information.
And I would echo that, Jeff. I think it also depends on like what our regulatory authorities are. And so when it comes to things like contaminated water, if that's a risk that could then contaminate food, EPA regulates the water, not FDA, so we would need to work with them in that particular space. So the different government agencies have different authorities. But in a One Health perspective, what that means is we all have to work together. And that includes USDA, that includes EPA, that includes us at FDA, but also CDC and others. So I think that's another thing we've learned through the pandemic is the amount of engagement we have had with other federal agencies. In the setting of SARS-CoV-2, that also included folks like the Department of Labor and OSHA, and we're working to protect farm workers and others who are at risk, both for themselves, their families, and their communities, but also that would increase the risk of food supply chain disruption. So the amount of engagement we had to mount during SARS-CoV-2 was certainly illustrative of the importance of working together in a One Health way when we have a public health emergency.
And I think Dr. Main's comments are spot on. We can always have conversations and tabletop exercises and those things, which is very valuable in preparation for emerging diseases and things like that. Obviously, in this last year, we've had one of the world's biggest tabletop exercises in terms of how we've dealt with this. And those lessons that we've learned and the times that we've had to work with these other agencies can be lessons that we can take forward.
Okay, great. Moving on to the next question. And I'm going to put a caveat on this one just because I'm a veterinarian. My wife is a small animal veterinarian. So I'm going to talk about something that's more cosmetic related. But just so people know, people use Avon products on their pets for insect repellent purposes. People put nail polish on their pets. People tattoo their animals for identification purposes. So there is probably more crossover here than a lot of people are aware of. But the question is this. The FDA is putting a lot of effort, a lot of resources into developing new methods or alternative methods, as we call them, to help reduce the amount of animal testing as it applies to food additives, packaging or food contact materials, cosmetic ingredients. So this is really sort of focused on Dr. Main and Dr. Camacho. How does this approach as far as reducing animal testing or increasing alternative methods, how does it fit in with FDA's predictive toxicology roadmap, and especially with the new technologies that NCTR is working on for safety evaluation for different products?
Well, maybe I can start and then Dr. Camacho can jump in. Yeah, the first thing I'll say, it's not unique to cosmetics. At the FDA, we really want to reduce the reliance on animal toxicity testing across the board, as you highlighted, for food ingredients, for anything that we regulate it. And the other point I'll make is that we want to get it right. We want to know what's the most predictive of human toxicity. And you can test compounds in 2 different species of animals and get 2 different answers. So it's not that animals always give us the best prediction. So I think what we're trying to do is really look at the totality of the science and use all of the tools that we have available. That may be in silico methods. What do we know about chemicals? What do we know about their structure? What do we know about what metabolic pathways they impact? What does that tell us about potential routes of toxicity? What in vitro assays do we have where we could screen large number of compounds at the same time? Are there non-mammalian systems that we can study that can really tell us something about toxicity? And then when we look at all of that evidence together, how do we get the best prediction about human toxicity risk? So some of the work we're doing in CIPSAN includes things like we're studying C. elegans. It's a nematode. It's a little teeny tiny worm. We're looking at that to see how that can model effects on neurodevelopment, neurobehavior. And these are, again, non-mammalian systems that could help advance much of the work, where the mammalian systems have been especially challenging to predict human toxicity in this area. We're also, like many other laboratories, working on microphysiological systems, organs on chips. And that is, can we do these types of methodologies looking at specific organs, whether it be lung or liver or gut, to really be very predictive of toxicity we would see in the human setting. So I think the whole thing is building the foundation of science so that we can do the best job possible predicting human toxicity, and it doesn't have to all revolve around animal models.
And I'd like to echo Dr. Main's words. So I agree, it's not just cosmetics, it really spreads across all the products that are regulated by the FDA, and really the effort to develop, validate tools that are alternative to the use of animals. I know it's a very important and critical effort by the agency, but again, we have to do it right and to build enough robust data that we can do the jump for those alternative methods. And so NCTR really has a lot of active research. As I mentioned, part of our mission really is to develop and validate innovative tools that can be used to support the FDA regulatory mission. And in all these projects, we work very closely with the colleagues from the product centers exactly to make sure that our work really goes towards their needs. And so in my talk, I mentioned one example of a study that we are conducting to try to assess whether bioprinted human skin can be a useful tool to replace the use of animal skin as well as human native skin for in vitro permeation testing. But we have, of course, other studies, including several looking at microphysiological systems such as placental, testes, liver, as well as also computational tools that can also be able to provide data without resource to animals. And so, between all these activities and the close interactions with the centers, we hope we can contribute to the implementation of the FDA predictive toxicology roadmap.
Okay, great. Moving on to the next one, which is a wide open sort of area to talk about. And I think many of you addressed this in your individual presentations. But, Dr. Main, Dr. Rice, Dr. Keneal, and I think Dr. Edwards, even you and CVM, who are using these approaches as well. Can you talk a bit more about the impact of the really revolutionary combination of whole genome sequencing, genomics, metagenomics, bioinformatics, as it relates to foodborne outbreaks and also in preventing and mitigating human or animal illnesses or deaths? And anyone who wants to start.
Sure, this is Dan Rice. Yeah, that's a great question. And so I remember I was moderating a session at the Association of Public Health Laboratories annual meeting about 10 years ago, and we had a panel of people. Eric Brown from Genome Tracker was there. Some of the PulseNet people were there. And that was back in the day of PFGE, pulse field electrophoresis, and we were talking about what is whole genome sequencing going to bring, what's it going to do for us. And And so now, 10 or so years later, it's a routine part of what we do. And there's really interesting data from CDC, the PulseNet group that I've seen that demonstrate the value of these technologies and identifying outbreaks quicker, reducing the size of the outbreaks. So right now we might have more outbreaks. It might seem like we have more outbreaks, but we're detecting more of them than we used to. And the overall number of cases is going down. And that was just with whole genome sequencing. And so, you know, who knows what the future is going to bring with metagenomics and the technology that's coming our way. It's just amazing how we just expand our capabilities exponentially. So I'm really excited to be in the area I'm in now and not close to retirement, so I can really enjoy what the future is going to bring. And I'll just leave it at that.
And I can give some perspective as well. I think, you know, when people think about outbreaks, we often used to think of it, you always start with the epidemiology, you figure out who's got the disease, use whole genome sequencing to link those cases together, even if they're geographically dispersed. Then you ask people what they ate, then you try to figure it out, and then you go test product following the epidemiology. But I think what we see now with whole genome sequencing is sometimes we'll find cases and we'll do the whole genome sequencing and we'll find that we have a cluster of illnesses and we'll look in the database and we'll have a clue to a food source before we've even done the, you know, the epidemiology piece of it based upon the sequence of the pathogen, because we've seen that pathogen before. We've seen it in a particular region. We've seen it in particular products. That doesn't give us evidence of causation, but it certainly gives us a head start in terms of trying to figure out what's going on in outbreaks. Another example I think is that we have learned that I think is really important for prevention is we have seen facilities that have produced contaminated food, and then we start sampling in the facility and we do environmental testing in the facilities and we find out they've got a pathogen in there and that's led to contamination. And then we go back, 6 months later to see how the facility's done following a contamination event. And we find the same pathogen again, and it's the same genomic strain, and then we know they've got a resident pathogen in there. That is a very, very serious food safety risk, and we want to address it. And our ability to find that and to know that they've had an ongoing problem with the same pathogen is really important in how we address prevention of future outbreaks. This is not a sporadic introduction of a pathogen. This is the same pathogen coming back over and over again. So we use that in the prevention piece of that. And in outbreaks, it's not just about getting contaminated product off the market. That's obviously, job one, but it's making sure it doesn't happen again. And whole genome sequencing is a tool that really can help us in that regard as well.
I think, too, one of the important pieces that we can look at with the whole genome sequencing is that preventive measure, where we maybe be finding that in a finished diet or in a pet food or something like that. But if you find that same pathogen and you've characterized it through some of these different techniques, you can trace that back into the ingredient and really find those other lots that might be a big part of And so we've seen probably more expanded recalls in the last few years because of this, because we are able to trace that back through. And it's not just, oh, it must just be this lot that's contaminated. You can also do a lot of that testing and figure out what things maybe need to be recalled before they even get out there, which I think has been, although it's hard to track some of that sometimes, it's been a real plus on our prevention of causing those issues and those pathogens from getting out into the general population.
So, Dr. Keneal, if I can put you on the spot, how is sort of the One Health area utilizing this part of science, the whole genome, using genomic methods for tracking things, or associating different things across the spectrum?
Yes, it's really critical in the development of the ease of implementation of not only the technology but also the data has really advanced over the past couple of years, and it's more available now across, again, private and public partnerships, not just federal labs, right? And I think that's really an important part of it. I certainly echo what Dr. Main said, which is so important is that prevention piece. And I've worked with producers who have had contamination events we are able to sequence those and show them that it's a repeated offender, so to speak, and that really helps with the educational component of that. I think to get to your question, Dr. Ward, it really, when we think about some of the research aspects that are happening, we can use whole genome sequencing and even some metagenomics to understand why certain organisms are in an environment. Is it just a niche where they're, very comfortable, they have the right nutrients? Maybe it's a sanitation issue if it's outside of an environment like a surface water, or is there a reservoir for that organism? Certainly we've had this discussion and debate even with salmonella for many years, where it survives in multiple environments and low moisture foods as well. Foods that we're not really certain about, and why do we have heat resistant microorganisms sometimes, but looking at the genomic analysis can really be very advantageous to help us understand why some of these organisms are there and relate them back to other situations. And again, as was mentioned, these databases that are so rich with these genomic sequences can be very helpful in helping us understand, where one organism was seen previously and now it's there. And I've been lucky enough, or I guess I feel fortunate to be able to work with the FDA scientists. Maybe the growers aren't so fortunate, but when we find an environment or that has salmonella, we find that organism, it's in Genome Tracker, and I get a call and saying, we found something that was just like that organism you found before. Can you tell us more about that environment? What animals were in that area? Where is there high poultry production there? Or were there reptiles? And so I think those discussions are really critical as we move forward to better understand, again, that One Health approach, what's going on in the environment. I think it's very exciting to see these tools really being used in so many different approaches.
Great. Very good. So the last question that we've come up with, and I might have to come up with more while you guys are talking since we're ahead of schedule. And this is I think really going to be a Center for Food Safety and CVM question, but I'm sure Dr. Rice, you can chime in, and Dr. Neal as well. And it has to do with the role of sort of intersecting or overlapping supply chains for human and animal foods. You've got byproducts or different components that go into both streams or that overlap. I think Dr. Edwards addressed, there are certain byproducts that come from the human food side that go into animal foods in some cases. So what role does sort of that intersection or interaction play in, for example, salmonella infection? And if something does come up, how are those issues detected and tracked, and can it be traced back to the other side? Meaning, if something shows up in animals, can it be tracked back to possible input into the human food supply side of things? So I'll leave that open to anybody who wants to respond.
I'm happy to start, Jeff, on this particular one, and I can give you one example where this kind of happened, and that was with the Peanut Corporation of America peanut butter outbreaks in 2008 and 2009. And so what we had was contaminated peanut butter that led to a lot of human food recalls, and then that subsequently led to recalls of pet food that was found to contain the peanut butter paste that was used in both human and animal food products. So the issue was first, found out in the human setting, but it uncovered a potential hazard for pets. And that is, when we find a contaminated product like this, part of what we try to do is figure out where the contamination occurred and then do like a trace forward analysis to see where that supply might have gone elsewhere to make sure that other relevant products are being recalled. And in this case, the human food setting led to animal, recall of pet food as well. And I'll say from an FDA perspective, CIPSAN and CDM work very closely together on these types of scenarios. Anytime there's a potential intersection between human and animal food, if we see an ingredient in human food that may have caused illnesses, if the supply chain leads to an animal food, CVM will be quickly looped into any of those kinds of discussions. So that's a real-world example where we've seen that kind of thing happening. So perhaps CVM wants to comment as well.
Absolutely, and I think that one of the things that we all know and something to remember is how closely tied together we are as a human and animal population. Whether it's our pets that live in our homes and the comfort that we give them and they give us, or whether it's the animals that are producing our food, we end up with a lot of the things on our plates having components that then also end up on the animal plate. So Dr. Main's example from the peanut, from the salmonella outbreak in the peanut products is a great example where we really needed to ensure that food safety is a big part of that One Health concept, that safety of human as well as animal health. And controlling those hazards in one part of these ingredients probably leads to safety of both humans as well as animals, because a lot of these components, I can think of many components of human and animal food that get split from the same essential origin. And so we see those things that way. The other component I want to mention here is that as we have things like industrial production, whether it's of ethanol and there are corn products that are coming from that, or other types of things, we need to keep those folks thinking about food safety as well. It's not only the production of whatever their primary product is, but it's the production of those co-products that is so important to both human as well as animal safety. We are lucky within FDA to have our folks in the Office of Regulatory Affairs aligned on both the human and animal health, animal food front. So those folks can very quickly be able to process through any problems that are happening on one side and think about how that occurs on the other side of that coin. And so I think that's one of the vital things. Obviously, the work together between our response teams in CISTAN as well as CVM is critical to this. And I think we, going forward, will probably have more and more overlaps as we are able to utilize some of the tools we talked about in the last question, whether it's the whole genome sequencing or others, to be able to prevent some of these outbreaks and hopefully find that in both the human and the animal front.
And the last point I'd like to add is, humans don't have to eat the food in order to get sick from the pet food. And Dave can speak about this issue with, contaminated animal foods, raw pet foods, things like that, and human illnesses that have ensued following that type of exposure as well. And you heard Dr. Keneal talking about the backyard chickens and things like that. So animals can contaminate, and including animal food can make people sick even if they're not ingesting it directly. So another potential link between animals, their food, and human illness.
Great. Okay, so now we're going to go off script a little since we still have 8 minutes to fill. So these will be questions that we haven't shared with the panel, but I think that's fine. I think just about anybody can chime in on this. So on the human side of things, the FDA has FERN, the Food Emergency Response Network, for handling food, for human food-related outbreaks. We have a laboratory network set up and a very elaborate response network. On the veterinary side of things, there's VetLearn, which is the veterinary lab network, which handles more animal-related issues. And I'm going to put Dr. Keneal on the spot here, and I know she's the one person outside of the agency. She's with University of Delaware, but I don't know that she has anything to do with how Delaware as a state responds. But the question is really centered around how do we interact, how do our federal guidelines and responses for outbreaks conflict or coincide or interact with state or jurisdictional specific responses to conflicts? Is that an issue? Is it still an issue? Is it something we're still contending with? And that could be from the animal or the human side of things.
Sure. I think I can speak to it a little bit from both, maybe, because at UD we work, my department works very closely with a VetLearn, perspective in terms of avian influenza and the poultry industry. And then, of course, I've worked with FDA a little bit when it comes to looking at human contamination issues in terms of foodborne illness. So I actually think there is a really nice interaction at the state level and with FDA. And I'll use the produce safety rule as an example. I commend the FDA for the term educate before you regulate. I really think that that's a wonderful sentiment. If there's another aspect that we've learned over the past year is that we cannot let up in terms of the science education that our country needs. We really need to continue to talk to people about how important it is to understand basic biology. And a lot of that is what happens during outbreak investigations. And when we think about disease transmission, I love what Dr. Edwards said earlier about the biosecurity. Just thinking about maybe color coding tools when you're moving from one field to the next, or keeping your truck in, off of one area from another if you have diseased animals there. There are things that, just basic issues about how diseases are transmitted and how we think about surveillance in our own fields. We need to continue to communicate. And I do think, from what I've seen with Delaware Departments of Agriculture, that I think FDA is doing a great job. And so I really think it's important that we have all of that multi-pronged approach from the federal and state levels, and we continue to have education as a component of that on all levels. Within our school systems, K through 12, it's never too early to really talk about the basics of science. And then in agriculture, teaching our children, wash your hands after you feed your dog. These are all good behaviors to start. And, and we really need to continue to have that discussion about backyard flocks, because they're growing in number and we don't want people to get sick, but we want people to love their animals. And that, I think, is difficult. That might be trickier than the state and federal communication.
Okay. We're just about out of time. I could bring up another subject, although I'm not sure we could cover it in 4 minutes. I'll throw it out there anyway. Antimicrobial resistance. That's a big issue that the agency often puts forward as a big One Health initiative. It's high on the list of WHO, OIE, all of these organizations, because it covers resistance of organisms in animals, in people, in the environment. Just thoughts, random thoughts. Are there gaps? Are there big issues that need to be dealt with there that we could make progress on? Especially, from the federal government to the state, even across the different agencies. And across the different centers of the government. Just throwing that out there. Maybe Dr. Kniel or Dr. Main, you could address that.
Well, I think CVM has done some work here in the use of these antibiotics in growth production in animals, and limiting those and trying to protect the use of the medically important animal antibiotics for human use. So, Dr. Edwards, I don't know if you want to just make a comment or two on some of the efforts CVM has been doing, because that has been one of the big areas of concern, is inappropriate use of antibiotics in animal agriculture. And that is work that FDA has been engaged in and has seen some progress. Dr. Edwards?
Sure thing. Found my unmute button too. So I think as we think about antimicrobial resistance, it is obviously a multi-pronged sort of issue that we have to work on. At Center for Veterinary Medicine, we have worked to ensure that there's the judicious use of those antimicrobials, especially those that are being utilized and are medically important on the human side, because we want to keep those useful for everybody, humans and animals alike. It doesn't help if we wear out antibiotics or have resistance to those by certain pathogens we're trying to take care of. In order to do that, we have worked with some of the sponsors that have come in, and at Center for Veterinary Medicine, as many of you know, we address both animal foods as well as animal drugs. On the animal drug side, we've worked, sponsors have voluntarily pulled the production claims from many of those antimicrobials. I think I mentioned in my talk too that with that change in how some of those antimicrobials are being used, there is additional research and interest in innovating to make sure that animals can stay productive and can continue to grow and produce those products that we eat. And so we have seen a lot of new innovative ingredients that are working in some of the similar spaces, trying to find where we can gain some of those additions to production in order to make sure that we have a healthy and safe food supply, and it's abundant enough for us in the future. So with that change and how a lot of those are utilized, we also have worked to make sure that veterinarians and health professionals are involved in when those antibiotics need to be utilized for treatment of certain diseases. And with that being put in place, that helps us to continue to work towards that One Health goal, to make sure that we have those tools in the toolbox to keep animals and humans safe. So it's a joint effort across many parts of both public and private, and we're excited about what the future brings with that.
Excellent. And with that, I'd love to keep talking, but thanks everybody who's tuned in to watch this this session, and thank you to all of our presenters. It's been an excellent session. I just wanted to thank everybody for attending the 2021 FDA Science Forum, and wanted to emphasize that we hope that you will all visit the 2021 FDA Science Forum virtual poster session exhibition, which is on the forum's webpage at fda.gov. So with that, again, thank you to everybody. It's been very productive. So everybody have a good rest of your day.